Hello, good evening students, good evening juniors. Hopefully you all are doing well with your exam and let's start, and please in the comments mention that I'm audible clearly and you can see everything including the things which are written on the board. Please let us know so that we can continue the session. And once your response will be there, then I will continue. Okay, good evening everyone. So first of all, बहुत छोटा सा अनाउंसमेंट है, हिंदी मीडियम वालों की क्लास एक दो दिन बाद होगी. So this class is for English medium, exclusively for English medium. So let us start. Now first of all, you need to know about the science and technology, how to study in last time. Got it? Last time means last minute strategy. So this class is going to cover at least 3 topics. Thank you, you people are waiting. And Achala Gupta, yes. So now I can recall also Achala, Dilip, Bhavika, Lalchand, Rajkumar, everyone. Good evening, Khushbu. Okay. So many names are I can recall and reconnect with the classroom students. So whoever is there. So now for next half an hour, kindly excuse me. I will not be able to respond to your comments and I will not be able to go for this thing. So we have to go through in a, you can say, way that exam is ahead. So we have to study like that. So let's start without wasting the time. Kindly excuse me if I'm not going to respond to your comments and I'm not going to answer your questions. Okay, so let's start. So first of all, what I'm going to cover today, I'm going to cover biotechnology. With biotechnology, we will cover blood groups, RH factor. With this, we are going to cover genetics also. First of all, we will go through this thing and then after we will go with the other things if the time and energy will allow me. Otherwise, we will continue tomorrow onwards. So first of all, biotechnology. Whenever you are studying about biotechnology, no need to go through the definitions of biotechnology, because right now our approach is prelims oriented. So first thing you need to understand is biotechnology term was given by Carl Ehricke. So first thing you need to cement in your memory is Carl Ehricke. Carl Ehricke was the man who coined the term biotechnology. That is enough for now. Then after, people will talk about biology and technology, merger of this branch, that branch. No one is going to ask you about the definitions. One thing. Second thing, who coined the term of biotechnology? Carl Ehricke coined the term biotechnology. Now let's jump to next field. There are different colors of biotechnology. Now, how it is possible that a subject is carrying colors? So whenever you are talking about the colors of biotechnology, it means they are talking about the application. And for your kind information, they are going to ask you only 2 things: basic concepts plus applications. So if you are aware of basic concepts, then only you can understand the applications. But in most of the exams, approximately 95% times the questions were asked from the applications. So now one by one we will talk about the applications of biotechnology. Obviously, before discussing the application of biotechnology, we need to understand the concept which is lying behind that application. So for example, when you talk about the colors of biotechnology, the first application is about the oceans. Whenever you are talking about the oceans, you need to recall Sir C.V. Raman. Whenever you are talking about Sir C.V. Raman, you need to talk about the Raman effect. The Raman effect, which is responsible for every launch of NASA, which is responsible for every launch of ISRO, which is responsible for the application of all type of waves, all type of, sir, please change the board color. Actually, I think it is visible. You people have shared a green signal from your side. That's why I have chosen this color. Okay. So whenever you're talking about the oceans, always remember anything about ocean is blue biotechnology. So in one line, you need to remember blue biotechnology means Yes, blue biotechnology means marine biotechnology. Anything about marine life, anything about ocean, anything about the application of biotechnology in the field of oceans is going to named as, yes, what? Blue biotechnology. Now, one application, most recent application. There is one product from ocean, that is chitosan. Now, it is my advice to all of you that please sit with paper and pen. It will be helpful for you because many things will be there. I will not be able to write down everything here because we are having very less time. One thing. Second thing, if you are having any problem about any of the basic concepts, you can come over to institution and you can meet me personally. I will sit there. I will spare my time with you. Got it? Now, Pallavi, this class is not for Hindi medium students. हिंदी मीडियम वालों के लिए क्लास 2 दिन 3 दिन बाद होगी, तो आप लोग अभी पढ़ाई कीजिए, अपना टाइम खराब मत कीजिए. ओके, सो नाउ वी विल टॉक अबाउट दिस थिंग. नहीं, हिंदी में नहीं होगा, ये वाला लेक्चर इंग्लिश में ही होगा. सो प्लीज हिंदी मीडियम वाले बच्चे बीच में कमेंट ना करें. Now let's start. So for example, there is one product of ocean named as chitosan. Now what is chitosan? So chitosan is used in wound dressings of army people. Now how it is useful? So yes, obviously whenever you are talking about wound dressing, it should be antiseptic. But with that, this is carrying extra property, that is anticoagulant. Now whenever I am speaking anticoagulant, the meaning of anticoagulant is anything which is going to stop bleeding is named as anticoagulant. So because of anticoagulant property, chitosan, which is a marine product, is nowadays in interest, in the periphery of interest of the scientists. Now why they are talking about this thing? Because crabs, you know very well that crabs are one of the examples of resources provided from the side of ocean. It is a gift of ocean. So crabs are the source of chitosan. One thing. Second thing, chitosan by nature is a carbohydrate. So whenever you will mix this carbohydrate, which is named chitosan, so I am going to summarize this thing. Why I am asking you that you are supposed to sit with a paper and pen, that all you need to know is C, C, C. C is from crabs, C is chitosan, and C is carbohydrate. And C is coagulation. Coagulation means it is going to stop the bleeding. So C, 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 C. Whenever you will say 4C, you need to say, yes, now I am carrying one example of blue biotechnology. Now, second example. Second example of, everyone is talking about bioalcohol or bioethanols. For your kind information, bioalcohols or bioethanols are used for mixing in or for mixing with the petrol. What is the benefit of mixing ethanol with the petrol? So the benefit is reduced pollution. How the pollution will be reduced if someone is going to mix ethanol? So you always remember ethanol, which is also named as C2H5OH. It is carrying oxygen molecules. Oxygen is an integral part of every molecule of ethanol. So whenever you are going to mix petrol with ethanol, it will lead to complete combustion of fuel. Whenever there will be complete combustion of fuel in the engine, there will be less pollution. Now, how this thing is linked with blue biotechnology? So nowadays, scientists are working with the algae. You always remember study of algae is named as phycology. Now they are studying about the algae. What they are studying about the algae? They are trying to modify the algae biotechnologically, modified algae, so that this algae can produce more bioalcohols. So that's how, thank you, that's how, सर एक कमेंट यहाँ पे आप पेस्ट करवा दीजिए कि हिंदी मीडियम की नहीं है ये क्लास, हिंदी मीडियम वालों की 2-3 दिन बाद होगी, जब होगी तब शेड्यूल अपडेट कर देंगे। वो बच्चे बार बार कमेंट कर रहे हैं तो उसको पिन करवा दीजिए थोड़ी देर के लिए। Okay, so this is blue biotechnology and there are 2 applications of blue biotechnology. Then next is green biotechnology. Whenever you are talking about green, you need to understand we are talking about plants. So now green biotechnology is very important by the point of view of your exam. Why? I'm going to give you some of the examples and these topics are going to be covered in today's lecture. First thing, BT crops. Second thing, out of BT there are BT advanced crops. Third thing, all type of GM crops. Yes, obviously BT crops are also example of GM crops. Then after, fourth thing, in green biotechnology, tissue culture. Then after, fifth thing, all type of genetic engineering which is associated with plants or genetically engineered plants. Then after, fifth is plant molecules. And their use in the field of, you can say, breeding or in the field of medicine or in other fields. So that's how green biotechnology is. हिंदी में क्लास नहीं है, प्लीज हिंदी मीडियम वाले बच्चे बार बार कमेंट ना करें। ओके, तो आपकी क्लास 2 दिन बाद होगी। तो आपकी क्लास में आपने कभी देखा है इंग्लिश मीडियम वाले कमेंट कर रहे हो कि इंग्लिश में क्लास हो रही है क्या, इंग्लिश में क्लास हो रही है क्या? गॉट इट? नाउ नेक्स्ट इज रेड बायोटेक्नोलॉजी. So whenever they are talking about red biotechnology, that means they are talking about the application of biotechnology in the field of medicine. Now whenever you are saying medicine, that means it is going to cover the entire field of healthcare. So anything which is associated with healthcare is going to be covered in the red biotechnology. Now in this we are going to cover many of the examples which are important for today. Why I have selected this topic for the first class? Many of the students, they consider this topic is very difficult, but according to me this topic is very useful for the exam. For example, what are the things which are covered in red biotechnology? Vaccines. Whenever you are talking about vaccines, you know very well that COVID-19 vaccines, they are always in the exams. Then after, second thing is enzymes. All type of enzymes which are produced. Then after, third is antibiotics. All type of antibiotics, they are produced under the periphery of, within the periphery of red biotechnology. So these are the examples of colors of biotechnology. But if someone is going to ask you in the exams that what is white biotechnology? White biotechnology. See, the marker is missing some of the words. That's why I asked you people to sit with paper and pen. So white biotechnology is applications of biotechnology in the field of industries. Anything which is happening on industrial level, industrial scale, with the help of applications of biotechnological techniques will be covered under the topic white biotechnology. Then after, If someone is going to say, what is yellow biotechnology? Yellow biotechnology. Then you have to say yellow. You need to remember only one thing, insects. Anything which is related to insects plus applications of biotechnology, that is going to covered by under the topic yellow biotechnology. Then one more thing which is very important nowadays, this question has been asked many of the exams, gray biotechnology. So if anyone is going to ask you what is gray biotechnology, you have to say applications of biotechnology in the field of environment, or I can say environmental biotechnology. It is named as gray biotechnology. Now one by one we will talk about all the aspects which are important for your exam. So let's start. I think so far everything which I am writing here is visible to you. I need at least one yes from your side so that I can continue. Because in mid of the lecture there were comments like please change the board of the color and like that. So if you people are okay with this thing, then I should go ahead. This class will cover environment also. Yes, many of the aspects of environment will also be covered. See, all of you are very well aware of one thing, that we are not having so much time. If anyone is saying that I can cover all the things in one class, it is impossible. One thing. Second thing, I cannot go through topic by topic. So what I am going to do, after so many yes, now I am going to go through this thing in such a way that we will only deal with the topics which are associated with the questions. Okay. So now let's start. So first thing which is very important nowadays is three-parent baby. Three-parent baby. Now what is three-parent baby? Again I'm saying I'm going to deal with this thing by the point of view of prelims. So now excuse me for next 5 minutes. After the completion of topic, I will give you a time to write down the comments. Okay. So now let's start. So no comments for next 5 minutes. Three-parent baby. What is three-parent baby? First thing, genetically, if the baby is carrying DNA of more than two parents. Now you will say how it is possible that someone can carry DNA of more than two parents. So now see, you need to understand the science behind that. DNA or chromosomes. Whenever you are talking about these things, that means you are talking about red biotechnology or healthcare biotechnology. All the information which is associated with our genetics or genetic code, they are lying in your DNA. So you are typically carrying a mindset that half of your DNA is result of your mother and half of your DNA is result of your father. Now see, mother and father, yes, I agree, they are giving you half of the DNA. But you need to understand one more thing. When you are talking about the reproduction, this is egg. This is nucleus of egg. And here is mitochondria. And within mitochondria, you need to understand one more thing. There are finger-like projections which are named as cristae. And here is DNA. This DNA is named as mitochondrial DNA. You need to remember this thing. People who are not from science background, they need to understand this thing very clearly, that reproduction is all about half you will get from mother and half you will get from father. But when you are entering into the field of biotechnology, I will say from the side of mother, half plus some more. This some more, what is this some more? This some more is mitochondrial DNA. And plus this half you will get from your father. So what the father will provide? Now one was asking me that will this class cover environment also? This class will cover a lot of topics. For example, right now see here. So what your father is giving? During the process of reproduction, the cell which is most important is sperm. Now you know very well that sperm is carrying a nucleus. A nucleus is carrying chromosomes, 23 chromosomes. Now, other than this nucleus, nothing is going to be provided during the process of fertilization. Egg will encounter with the sperm, and as a result of this, zygote will be formed. Now, here you need to go through 11th NCERT chapter reproduction. If you'll go through that, you will come to know that egg is covered with the layers. This is corona radiata. The inner layer is zona pellucida. And inside this, there is nucleus. Other than this, there is mitochondria. And here is mitochondrial DNA. Mitochondrial DNA. In case of sperm, the sperm will only give the nucleus. So as a result of this, N plus mitochondrial DNA, that's how the zygote will be formed. And as a result of this, zygote will carry 2N, and with this there will be mitochondrial DNA. Now here you can understand this mitochondrial DNA is gift of egg cell. That means mitochondrial DNA is coming from the side of mother. Now just imagine if there is any defect in mitochondrial DNA, then it will be copied as it is into the offspring. So this may lead into, this may result into the problems which are genetic in nature. So if mother is carrying any kind of defect in her mitochondrial DNA, it will be transferred as it is into the DNA of offspring. Example of this is, there is a problem named, this board is not working properly, leg syndrome. Leigh, leg syndrome. In case of leg syndrome, it is a defect of mitochondrial DNA. That means this disease is a disease for which I can say that mother will give this to the offspring or to the child. Now, as a result of this disease, the dangerous thing is, before the age of 12, child will die. How the child will die? Due to failure of neurons. Now why the child is dying? The answer to that is, child is dying due to the defect in the mitochondrial DNA. And from where the mitochondrial DNA is coming? You will say from the side of mother. Now you know very well that to get a child, you need a sperm and an egg. It is nowhere written that you need the mitochondrial DNA of the same mother. So what is three-parent baby? The solution to this problem is the three-parent baby. So what we are going to do, we are going to take the egg of mother. We are going to take mitochondrial DNA of a healthy female. This healthy female is not the mother of kid. So mitochondrial DNA from this lady, nucleus of egg from the mother, and nucleus of sperm from the father. These three things will result into formation of three-parent baby. And always remember, UK was the first territory which legally said that yes, this technique is allowed in our hospitals. Now from my side, this topic is done. Any problem, then please let me know. Otherwise, let me proceed to the next one. I'm waiting for your yes. And please try to understand, I need to go in such a speed because we need to cover a lot of topics. So that's why I'm not wasting my time into a drawings or something like that. Okay, so now let me start with the next topic. Next is genetically engineered insulin. So genetically engineered insulin is what? See, whenever you are talking about insulin, you need to understand two things. One is, this is essential for all the patients who are suffering with diabetes. By the way, meaning of diabetes is, whenever you are talking about diabetes, you need to understand one more term. Many times they have asked, what is hyperglycemia? Hyperglycemia means increased sugar. Sugar means glucose in blood of patient. One thing. Second thing is, whenever there is hyperglycemia, there will be delayed wound healing. That's how I said that a lot of things are going to be covered here. Okay, now let me sit, please. Okay, so delayed wound healing will be there, and one more thing will be there, that is eyes will get affected. So the patient may face problems related to eyesight. Now, whenever you are talking about hyperglycemia, the treatment of this is insulin. Now, insulin is the hormone which is produced from the organ which is named as pancreas. Pancreas. Whenever you are talking about pancreas, you need to understand one more thing, that pancreas is somewhere near your stomach. It is something like this. There is head, there is tail, there is body part, and within pancreas there are different type of scattered cells. These cells are named as islands of Langerhans. Langerhans was the first person who Langerhans was the first person who discovered these cells, and these cells are of three types: alpha cells, beta cells, and delta cells. Now the product of alpha cells is glucagon. Product of beta cells is insulin, and product of delta cells is somatostatin. All three are hormones, so they are studied in the chapter which is named as endocrine system. Now there is a mechanism which is going on in your body. Glucose, whenever it is in excess, it is converted into glycogen, and with the help of insulin it is converted into glycogen, and glycogen is stored in your brain, glycogen is stored in your muscles, and glycogen is stored in your liver. Your liver is capable to store approximately 100 milligrams of glycogen. So your liver is a storehouse of glycogen also. Formation of glycogen from glucose is named as glycogenesis. One thing you need to remember, this step is responsible for healthy living of me and you. If this is not happening, that means somewhere the level of insulin is compromised. Whenever the level of insulin is compromised, the only thing which can replace insulin is insulin. Now, this thing is going to be a bit technical, so please keep yourself aware from everything which I am going to say in the upcoming 4 or 5 lines. Now, I am saying the only substitute of insulin is insulin. Now, the source of insulin. Now, what is the source of insulin? The only source of insulin was animals. Animals which are mammals. All the animals which are carrying, you can say, pancreas. From these animals, you can get insulin. But there is a problem with the insulin which is, you can say, collected from the animals or after slaughtering the animals. Now, what is the problem with the animal source of insulin? The problem is the peptide chains. Now, when you are going to study the structure of insulin, insulin is actually a hormone, and whenever you are saying this product is hormone or enzyme, always remember, other than sex hormones, all the hormones are made up of proteins. And whenever you are saying that this thing is a protein, that means proteins are made up of amino acids. And the chains of amino acids are actually leading to the peptide chains. Now, there are 3 types of peptide chains in the insulin. So, peptide chain A, peptide chain B, and peptide chain C. Now, just imagine the black one is peptide chain A, the green one is peptide chain C, and the other black is also peptide chain B. Now, whenever beta cells are going to produce insulin, the insulin is actually produced in the form of proinsulin. Now, what is this proinsulin? Proinsulin means this is A chain, this is B chain, they are linked together, and there is C chain also, like this. But when our body is going to utilize this insulin, the proinsulin will get converted into insulin. And in the resultant insulin, there will be only chain A and chain B. So our body is habitual to use this insulin. But when you are going to slaughter an animal and you are going to get insulin from the body of that animal, you will get proinsulin in place of insulin. So as a result of this, if I am going to inject the insulin from the animal source into the body of any patient, that patient will receive unnecessary chain C. And whenever you are going to receive anything which is unnecessary, your body is going to act against it. So as a result of this, this animal source insulin that is actually proinsulin, that means A plus B plus C chain is going to create allergic reactions in the patients. So the patients are going to face a lot of problems. Now the solution to this problem is we are producing biotechnology is producing chain A and chain B, peptide chain A and peptide chain B. They are produced separately. How they are produced, we will understand in upcoming lecture or in upcoming topics, that is about recombinant technology. And then after, they are going to join this A and B. They are producing A in a separate layer, B in a separate sample, and then after they are joining it. So what they form? They formed insulin. This is the science behind genetically engineered insulin. In 1983, 1983, Eli Lilly, was the company of USA, biotechnological company of USA, which was first one who discovered this, who invented this technique, or who developed this technique. And in India, there is a very famous company named Biocon India. Now, I'm done with one more topic. Kindly let me know you are okay with this or not. Now, when I'm saying kindly let me know, that means if you, any one of you is having any problem or any question, they can also share their comment. And always remember one more thing. These chains are joined by the help of disulfide bonds. Disulfide bonds. Harf e dil. Wow. So nice name. Priyanka ma'am, okay. What about others? Any other comment? Okay, Kuldeep is saying okay. Now, next thing which you need to understand is GM crops. One of the very important topic, GM crops. Okay, I'm not asking you people for the recommendations that what I should teach. I'm just asking you, you are done with this topic or not? That's all I'm asking. Yes, good evening everyone. Yes, we will cover everyday science also. Don't worry. Okay, now see, GM crops. Whenever you are talking about GM crops, what you are in need to understand on the prelims level. First thing is genetically modified crops. Whenever the genome of any crop. Now, many of you can ask what is genome. So the set of all the genes of any organism is named as genome. So when the genome of any crop is altered in any way, that crop is named as GM crop. So first thing they are going to ask you, which crop was the first GM crop of world, which was approved for the use? So always remember it was a tomato. Flower saver tomato. Good evening everyone. Flower saver tomato. That was the first GM crop. Then after they are going to ask you, what was the specialty of this GM tomato? So you need to understand one thing, that this tomato was not BT cotton. So the first GM tomato, the specialty of this GM tomato was it was carrying extended shelf life. Extended shelf life means whenever you are going to store it, it will not convert it into a rotten tomato very soon. The time or lifespan of this tomato after harvesting the tomato was increased. How it was increased, no need to dig into that. Then after they are going to ask you, which crop is legally authorized by the authorities, or legally they said that yes, you can use this crop in India. Then you have to say the only crop which is approved is BT cotton. Now a lot of things are happening about BT cotton. It is important by the point of view of NCRTs, it is important by the point of view of basics, and it is important by the point of view of current affairs also. So next we are going to discuss about is BT cotton. So first of all, what is BT? BT stands for a bacteria. The full form of BT is Bacillus thuringiensis. You need to keep this thing in your head, Bacillus thuringiensis. No need to mug up the names, but at least you should revise it 2 or 3 times so that you can recall it when you're sitting in the exam hall. Because the easiest exam is prelims. This is the only exam in which there will be question and they are giving you answer also. So if you have, you can say gone through it 2 or 3 times, you can easily hit the question. Now see, Bacillus thuringiensis is a bacteria. Now what is the specialty of this bacteria? BT is a bacteria and this bacteria is capable to produce special type of proteins. Now, what is the special thing about these proteins? These proteins, when these proteins will interact with the GI tract of insects, they will get converted into crystals, and these crystals are capable to perforate through the GI tract of insects. As a result of this, insect will die. Just imagine that someone is, you can say, sitting there and waiting to eat up the cotton or the crop, and जैसे ही वो पहला bite करेगा, this protein will start to react, and this protein will get converted into the needle shaped crystals, and these crystals will make a perforation. They will make holes in the GI tract of that insect, and that insect will die. Now, scientists are aware of one thing. If I am carrying any quality, that is because of my genes. That is because of my genes. If this bacteria is carrying this quality of forming this protein, that means there should be a gene system in this bacteria. So when they read the genome of this bacteria, they find out the cause of production of this protein is CRY gene system. Sometimes people call it chrysine. They should not call it chrysine. They should call it CRY gene system. Now, they will isolate this CRY gene system. Now, this CRY gene system is isolated. And after isolation, this will be inserted into the genome of crop. So what will be the result? The result of this thing will be, now this is the crop's genome, the black one, and this is CRY gene system. Now, what if someone is going to add this genome or this set of gene into the genome of crop? So you have altered the crop. That's why it will be termed as GM crop, genetically modified crop. Because this CRY is taken from the source, which is Bacillus thuringiensis, so that's why this crop will be named as BT crop. Bacillus thuringiensis crop. So Bacillus thuringiensis crop is carrying a set of gene which is named as CRY gene system into the genome of this crop. As a result of this, whenever there will be growth of plant, this is carrying CRY gene. So when the insect will eat up any part of this crop, the CRY gene system will be activated. It will produce the protein which is named as BT toxins. And as a effect of this, that's why BT brinjal is not approved. Yes, we will talk about this also. That's as a result of this, the BT toxin will kill the insect, and insect will die immediately. And as a result of this, you can say that you have prepared one of the crops which cannot be eaten up by the insects or by the, you can say, pests. So now next thing is, if it is sir, last time many of my questions were corrected and I had cleared RAS prelims. Oh, good luck for this year also. Good luck to everyone. Okay, now see. So as a result of this, you have created a crop which cannot be, you can say, damaged by the pests. This is what the science behind BT crop. Now, if someone is going to ask you what is terminator gene. Now see, obviously this crop is result of research. So whenever they are investing in the research, they are expecting a good return. There are no free lunches in the world. If someone is sitting there and thinking that they will prepare a crop or they will prepare a GM variety of some crop and then after they will give it for the social cause or for the welfare of the world, no, they will not give you. These are the castles in the air. Castles in the air are not going to work practically. So the company needs a good return. And when the company will get a good return, when farmer will purchase again and again the same seed. So what is the trick behind this business? The trick is terminator gene. Now what are terminator genes? Just imagine I'm the farmer and I have purchased the crop seeds against X rupees. Now next time I will try to produce my own seeds in my own field. I will say why I should go and buy new seeds every time. I will harvest the crop and I will keep a part of that crop for the purpose that they will be used as a seed in the next generation. But the company have introduced one type of genes, they are named as terminator genes. Now what are terminator genes? As you know, terminator is the term which is used for anything which is going to stop any process, any chain. So now, because of entry of termination gene, seeds will not produce any product in future. Yes, there will be seeds, but these seeds will never convert into a, you can say, produce. So as a result of this, you can say terminator genes are a kind of sterilization. Not exact sterilization of the plants. So now from here you need to understand why we are against the BT crops. Number one, these crops are going to establish the monopoly of the companies which are carrying the patent. Number two, what will happen if this terminator gene will enter into other type of plants? And third thing is, because of entry of BT crops, the local varieties are going to be eradicated. They are not going to diminish, they are going to be eradicated from the system. That's why our government and Bharatiya Kisan Union, they are against BT crops. The only BT crop which is allowed in India is what? Yes, that is BT cotton. Now, there is one current affair related phenomena, that's what? Pink bollworm. Pink bollworm is the answer of comment of one of the students who was saying that the insect will adapt according to it. So pink bollworm is showing this is resistant for BT crops, BT cotton especially, and it is causing a lot of damage in the areas of Maharashtra, Gujarat, and in Rajasthan, Ganganagar, Hanumangarh, Bikaner. These areas are the areas which are producing, you can say, cotton. And the farmers are very much, you can say, terrified by the effect of pink bollworm. Against pink bollworm, recently, state of Gujarat and Maharashtra, they have filed an application. Now, what type of application it is? This application is filed into the Office of Authority, which is itself a question of GK. The authority is GEAC. Now, what is GEAC? Genetic Engineering Appraisal Committee. Always remember, in India, all type of GM crops, all type of genetic engineering is approved by GEAC. Now, they have filed an application that please say or please authorize us for use of advanced BT. Now, what is advanced BT? Advanced BT is going to kill this pink bollworm. Now, this advanced BT is carrying CRY2AI gene system. CRY2AI gene system. And again, the seed is costier than the previous version of the seed. From here, there is a chain of questions we are not going to enter into, because we are supposed to finish with the point of view of prelims. So very soon there will be new BT, and this BT is pending for approval. And Gujarat and Maharashtra government, they are requesting that please say yes to this BT, CRY2A, CRY2AC, CRY2AB. These are the different genes which are associated with BT system. So I think we are done with BT. One more topic, BT. GEAC was a subtopic, Genetic Engineering Appraisal Committee. If any question, then ask here, or if you're okay, then say yes. I will go ahead. I'm waiting for your comment. Oh, GEAC under which department? GEAC is working under the Department of Environment. Ministry of Environment is the ministry which is Environment, Forest, Climate Change. In India, it is named as, right now in current government, Ministry of Environment, Forest and Climate Change. This is the name of Ministry of Environment. Okay, so hopefully we are done with BT. Now next is, we are going to talk about one more crop, that is BT brinjal. Whenever we are going to talk about BT brinjal, always remember that BT brinjal is not approved by GEAC in India. Now, it was not approved by GEAC in India, but recently it was in news. Why it was in news? Because in Bangladesh, they are growing BT brinjal from 2013. One reason. Second reason is BT brinjal was in news because in Indian market, most of the brinjal which is available is actually BT. If you will try to recall, in last 5 or 6 years, sir, screen half hai. How it is possible screen half hai? Is there any problem with the screen? Let me check it. No, I don't think there is any problem. Everything is clearly visible. I have checked it myself. So the only I was saying that you need to try to recall after you can say in the span of last 5 to 6 years, you can see the size of brinjal is increasing in India. So all that is BT brinjal, and how without approval farmers are growing it. So the thing to worry about is there is no facility, there is no chain which will go into the fields, which will come into my field, and they will say that which crop you are harvesting. No one of us is bothered about it. Okay, so you need to understand this thing. One more thing which is supposed to be retained whenever you are talking about BT brinjal is MICO. Now what is MICO? MICO is a company named as Maharashtra Hybrid Seed Company. Maharashtra Hybrid Seed Company. This is the company which is responsible for the development of BT brinjal. Now BT brinjal is collectively developed by MICO with the help of Dharwad University, the scientist of Dharwad University. I think promotion, BT, Brenjal, they didn't introduce. Yes, they did not. Done. So next thing we are going to discuss is, so now I'm going to end up the chapter topic of BT here. So we are done with 3 or 4 topics in 1 hour. So with the same pace, I will continue. Now, if you are okay till now, then please mention yes, so that I can move to the next topic. Let me hydrate myself. Only one yes. There are 60 plus students who are watching. Please stay with me with copy and pen. This session is going to be quite useful for you, quite productive. And I'm going with a pace in which if you will read it today only, then even then you will say that, yes, I'm done with this topic. All you need to do is you have to write down a lot of things you need to write down. Otherwise, it will go over your head. And deliberately, I'm telling you, deliberately, I'm not mentioning the technical terms here, so that you should be aware that you need to write down. Okay, Achala Gupta and Muskan, a lot of students. Gitika, okay. Achala Gupta is my student, I think. Okay, so next topic is DMH 11 or DMH 11. So first thing you need to remember about DMH is DMH 11 was discussed in Parliament of India. When it was discussed in Parliament during the monsoon session, Science Minister talked about it. Why he talked about it? Because he was questioned. Why he was questioned? Answer to that is DMH 11 is a variety of mustard. This is the variety of mustard. So we can say, what is DMH 11? DMH 11 is GM mustard. Now this GM mustard is undergoing field trials. This is undergoing field trials. Now, because this is undergoing field trials, so the Science Minister of India was questioned by the leader of opposition or some leader of opposition that yes, Deepak Patel, they were asked that they were asking what are the results of field trials of GM mustard. So the answer of Science Minister was, we are getting very nice results from the field trials of GM mustard. GM mustard, genetically modified mustard. So first of all, now we need to understand what is DMH 11. D stands for Dhara, M stands for mustard, H stands for hybrid, DMH. Now why 11? This 11 stands for the gene system. Now this 11 stands for the gene system. Now what is gene system? Now which gene system here is? So first of all, what is the use of this Dhara mustard hybrid and who have prepared it? So Dhara mustard hybrid or DMH 11 is prepared by a well-known professor of Delhi University. The name of professor is Professor Deepak Patel. Keep this thing in your head. Professor Deepak Patel. He is the developer of this crop, which is named as DMH 11. Second thing, DMH 11 is world's first GM mustard. This is world's first GM mustard. One more thing you need to remember here, mustard is scientifically, mustard is named as Brassica campestris. The scientific name of mustard is Brassica campestris. Now, what is DMH11? 11 is what? Bar, Barnase and bar star. You need to remember this thing. Bar, Barnase and bar star. Now, these are the subtypes of bar system, Barnase and bar star. These are the gene systems which are acquired from a soil bacterium or soil bacteria. Name of bacteria is Bacillus amyloliquefensis. From this bacteria, Barnase and bar star gene system is obtained, and then after this gene system is mixed into DMH or Dhara mustard hybrid. So if someone is going to ask you what is Dhara mustard hybrid, your answer should be this is the result of insertion of bar system. Now bar system, bar star and Barnase. What is the benefit of insertion of this gene system? The result of this gene system is the produce in terms of oil yield, because this is an oil crop, was more than the conventional mustard. First of all, it was examined in the controlled area of laboratory. Then after, it was approved by GEAC for the field trials. Field trials means the scientist will select the fields of mine, your or someone else, and then they will sign a contract with the farmer that we will take care of everything which is going to happen in this field. So they will cover the entire field with the poly house. And then after they will harvest the crop, they will collect all the things. Every grain of that crop will be collected by the company or by the contractor. And then after they will produce this data in front of GEAC, Genetic Engineering Appraisal Committee. Always remember, still this crop is not approved by GEAC. But this is, you can say, in news. So any question about DMH11, then please ask. I'm waiting. I'm waiting for yes from your side. As I said, you people are supposed to stay actively with me. Okay. So next is GM rubber. GM rubber. Hopefully, every topic is covered within 5 or bar system is took from bacteria and then it leads to more oil. Yes, exactly. One more purpose of insertion of bar and Barnase is to keep the plants sterile so that they should not pollinate in a cross way. Okay, so next is GM rubber. So first of all, you need to understand one thing, that rubber, GM rubber, whenever you are talking about GM rubber. Now, 3 or 4 things of science I will discuss about rubber here. Then after I will talk about GM rubber. And everything which is new to you, you need to note it down. So whenever you are talking about rubber, rubber is example of natural polymer. Natural polymer. Now, whenever you are talking about rubber, from where you get the rubber? The source of rubber is rubber plant's stem. From the stem of rubber plant, you get a milk-like structure which is named as latex. So you need to remember this thing. This latex is converted into rubber, which is named as natural rubber. Now natural rubber is the polymer, so the monomer of natural rubber is also important. The monomer of natural rubber is isoprene. So natural rubber is polymer of isoprene, or the government approved the release of DMH 11. No, it is not approved yet. It is undergoing field trials. Okay, so the monomer of natural rubber is isoprene. One more thing you need to understand here is rubber. Whenever I am saying rubber in this topic, that means natural rubber. When it is mixed with sulfur, the sulfur will form bonds between the chains of isoprene. This is one chain of isoprene, this is other chain of isoprene. Because of this, it will lead to hardening of rubber. This process is named as vulcanization. Vulcanization. Got it? Now, rubber. Whenever you are talking about rubber, rubber needs two type of conditions. One is A type of climate. Now you know very well, whenever you are talking about the A type of climate in Köppen's classification, it is available only in these areas. So this area is the area where you are capable to get rubber. Why? Because the requirement of rubber crop is humidity plus temperature, high temperature also. Now you know very well that in this area, the average temperature always remains about 20. Now, in India, there is one more place where you can get highly humid environment. This area. The entire area of Assam, Meghalaya, Manipur, this area is also humid. But the problem with this area is, this area can provide you with enough rain. But the temperature in winter will go down. So how you will deal with this problem? So answer to that is GM rubber. There is a place named Kottayam. Kottayam is in Kerala. There are only two places in Kerala which is supposed to be remembered by you. One is Kottayam. Why Kottayam is important? Because in Kottayam there is Rubber Research Institute, about which we are talking. Now the scientist of Rubber Research Institute, second I will talk about later. Second I will also talk now. Now say Kottayam, the scientist of Rubber Research Institute of Kottayam, they have researched a lot about manganese superoxidase enzyme. This enzyme is responsible for temperature resistance in the plant of rubber. Any type of temperature resistance, if it is present in the plant of rubber, it is because of manganese superoxidase. So what they have done, they have prepared a GM rubber variety. How they have prepared the GM rubber variety? They have modified the levels of manganese superoxidase enzyme. As a result of this, now there is first GM rubber of world. Once again I am saying first GM rubber of world. And as a prototype, this project is used right now in the jungles of Assam. So right now they are growing it in Assam. So I think we are done with one more topic. Again, if you are okay, then say yes. Every 10th or 15th minute, we are supposed to done with one topic. Then only these classes are like useful for you. If I will start to do the things like foundation classes, then it will be quite difficult for you people. And yes, second is pending about Kerala. Okay, now in the field of science, second thing you need to remember about Kerala is that is Thiruvananthapuram. Now, what is special about Thiruvananthapuram? Whenever you are talking about Thiruvananthapuram, you need to remember turtles. Now, what is turtles? Turtles is Thumba Equatorial Rocket Launching Station. Plus, one more thing is there in turtles, in Thumba or in the area of turtles. This is what? This is VSSC, Vikram Sarabhai Space Center. Now, what they are doing from this turtles and why this is important? So, you need to understand that there are 4 type of things which is happening in ISRO. Number 1 is they are launching satellites. Number 2, they are launching probes. Third, they are launching dedicated missions. And fourth, they are launching sounding rockets. Satellites, probes and missions. Example of mission, example of probe and example of satellite. Example of satellite is anything which is launched under the name of GSAT, anything which is launched under the name of EOS. We will discuss about it in detail later on. Probe means whenever you are sending anything to other planet. For example, Mars mission. Dedicated mission means Aditya L1. And when you are talking about sounding rocket, that means you are sending a rocket to study the upper atmosphere. Always remember, first rocket was launched in November 1963 from Thumba. And the name of place from where it was launched is Turles. Now, why Turles is so important that I am saying you need to remember it. Answer to that is lying in the structure of Earth. Now, what is special about this thing? This is the structure of Earth. Everyone knows that this is North Pole, this is South Pole. But you need to remember one extra thing when you are studying space technology. Now, what is that extra thing? This is North Pole and this is South Pole, but they are geographic poles. What is the meaning of geographic poles? If you will switch on the compass of your cell phone and you will indicate the north of compass towards the north, which is north for you, then you have observed, you can observe one thing, that north will not indicate towards the north. It will indicate slightly deflected towards the northwest. If you are agree, then type yes. No need to type yes, you can type Y only. I will understand that you are getting the concept. And in the same way, when you are thinking that this is east, it will show that no, that is not the east. East is slightly tilted from the angle which is east for you. And same is applicable for all the directions. Now, reason behind that is this is geographical North Pole, but the magnetic North Pole or real North Pole is here. If magnetic North Pole is here, then magnetic South Pole is lying here. Now, this is magnetic North Pole and this is magnetic South Pole. If we will join the line from magnetic North Pole to magnetic South Pole, then there will be one equator, and this equator will be named as magnetic equator. Magnetic equator. When you will study the general science, you will understand one concept, that any magnet is having two poles, north and south, and magnetic lines, they will always originated from North Pole and they will come towards South Pole. And the density of these lines will be maximum on North and South Pole. In the same way, the magnet of Earth is lying like this, not like this. But we have not disturbed this system, because if we will say to the geography people that go and correct your maps, then what will happen to your favorite Oxford or Oriental Black Swan Atlas? And everything will become, you can say, jumbled in such a way, as you people were panicked during the announcement of new districts in Rajasthan, that how we will study the Ganeshwar civilization now. Yes, that will always be near the Kantli river. But what about the district? Is it Sikar or it is Neem Kathana? In the same way, the things will change drastically. Just imagine that Rajasthan in Kashmir, right now it is in north. But if you will start to implement this system of magnetic North Pole, the Kashmir will not be in north. It will be northeast. And northeast will become something else. So that's why space science is different from geography. So this is the magnet or magnetic equator of Earth. Now, that's how the magnetic lines of Earth are protecting Earth. I'm using term protecting Earth. Why I'm saying protecting Earth, I will tell you. They are defending you against the harmful solar ionic radiations. A lot of plasma is erupted from solar radiation, and they are trying to approach the Earth. But who is defending you? This magnetic aura of Earth is defending you. This magnetic meridian system is responsible for defense of Earth. Now, because of this, scientists, they have observed that this thing or the interaction of the magnetic field with the solar ionic emission is a result of different type of lights on the North Pole and South Pole. They are named as auroras. There are two type of lights, green light and red light. Why only two type of light? Because in our atmosphere there are two type of ions, they are maximum. One is oxygen and second is nitrogen. Now in comment section, you people are supposed to tell me, if I am watching green light, that is result of reaction of solar radiation with nitrogen or with oxygen. Now let's go towards this. Now this magnetic meridian or magnetic equator is going near through the Thumba. So that's why Thumba is named as TERLS, Thumba Equatorial Rocket Launching Station. Yet the equator is not passing through the Thumba, but magnetic equator is passing from here. That's why it is named as magnetic Thumba Equatorial Rocket Launching Station. Now from Thumba Equatorial Rocket Launching Station, the rockets which were launched by India were Rohini sounding rockets. Rohini sounding rocket is mentioned like this. And with this, they will always mention a numeric value. This numeric value is diameter of rocket. So RH-75, RH-100, they were used in past. The first Rohini sounding rocket was launched in 1965. And recently, ISRO have launched 200th sounding rocket. But remember one thing, nowadays sounding rockets are launched from Thumba as well as from SDSC Shar, Satish Dhawan Space Center, Sriharikota. From there also they are launching the sounding rockets. So now we are done with GM rubber. One more topic we are done with. If they are going to ask you anything about this equator or something like that, you will answer, I think. Oh, Dr. Vikas Chaudhary is with us. Welcome, Vikas sir. Welcome. Okay, so now next we are going to discuss about is ART. ART in the field of biology is named as assistive, R is reproductive, and T is technology. In the field of assistive reproductive technology, the first thing you need to remember is anything or techniques which are helpful in treatment of infertility. Now, many of you are sitting there and they may be, Vikas sir, geography marathon please. Yes, exactly the same I was about to say. That student are going to demand the class of Vikas sir. Now see, anyone who is sitting there is aware of one thing, that there are occasions when they have asked questions from test tube baby, they have asked questions from stem cells, they have asked questions from surrogacy. So all these topics are covered under ART, assistive reproductive technology. So right now, first of all, let me tell you the agenda of this topic. First of all, we will cover sperm donation. Then we will cover IVF or in vitro fertilization. Then after, third thing we will cover here is surrogacy or surrogate mother. Fourth thing we need to cover is clone. And then after, we will cover other advanced technologies which are mentioned under ART, assistive reproductive technology. So let's start with one by one. There are two terms you need to remember. One is oligospermia. One is azoospermia. Now, oligospermia means less sperms. Azoospermia means zero active sperms. When I'm saying less sperm, that means I'm talking about sperm count less than 20 million sperms per ml. When I'm saying sperm count, that means I'm talking about healthy sperms. When I'm saying healthy sperm, that means any sperm which is carrying two qualities. One is sperm can move or movement, and second is the sperm is carrying a healthy nucleus. Whenever you are talking about the sperm, you need to understand three things. The head part of sperm is carrying enzymes. This part is named as acrosome. You need to understand this thing, acrosome. Acrosome is containing enzymes. These enzymes are hydrolytic enzymes. Hydrolytic enzymes will help the sperm to rupture the outer layer of egg. Outer layer of unfertilized egg, which is named as zona pellucida. Zona pellucida. And other than zona pellucida, there is corona radiata also. But here is the nucleus of unfertilized egg. So this nucleus is supposed to merge with the nucleus of sperm. Second thing you need to remember is the middle part of sperm is containing a lot of mitochondria. These mitochondria are responsible for movement of sperm. And then after, there is tail. This tail is helpful for the sperm to float within the genital tract of the female. Now, whenever you are talking about sperm donation, always remember that sperm donation as well as surrogacy, both of them cannot be commercialized in India. They cannot be commercialized in India. So always remember, in the movie Vicky Donor, you might have seen that he is purchasing a lot of things by means of selling his sperms. It is not possible in India. It is legally a crime if someone is doing this, one thing. Second thing, for surrogacy, always remember, the people who are studying in Hindi medium, they will keep on calling it kiraye ki coke. or in English I can translate it something like womb for rental. Always remember, even if you are calling it like this is illegal, and you will be shocked. People are studying it like that. That there is something named kiraye ki coke. No, you cannot call it. You're not allowed to name it as kiraye ki coke. Why you are not allowed to call it kiraye ki coke? We will discuss it. Now, sperm donation means in case of oligospermia and in case of azoospermia. There are two different ways. In case of oligospermia, the patient can go through the process like IUI or IVF. But in case of azoospermia, the only chance is sperm donation. So they can take from BSc में यही बोलते थे. No, they cannot call it. There is act named as Surrogacy Technique Regulation Act 2019. It is passed by the Honorable Parliament of India. And this question was first of all raised by one of the very prominent lawyer of India, very knowledgeable lady, and I pray for her to rest in peace, Honorable Sushma Swaraj. She was the first one who raised this thing in the parliament in an effective way, that this technique is used negatively worldwide. We'll deal about this also, but after every 10 or 15th minute we have to done with one topic. That's why kindly excuse me, we will discuss it later. Now see, what is IUI, what is IVF? Now sperm donation, that is enough for prelims. Now IUI is intrauterine insemination. Now what is intrauterine insemination? Simple, 3 steps you need to remember. Collect semen from male, first step. Second, sperm or semen treatment. And third, introduce the semen or sperm sample into female. So this will increase the chances of fertilization. How this will be performed? So you need to understand one thing from general science, and I will tell you many questions from this. This is vaginal canal, this is uterus, and this is fallopian tube. Here is ovary. This is unfertilized egg or ovum. Now from here the egg will be released or the ova will be released, and then after it will reach here. Now in case of normal process of reproduction, the sperms will be sprayed here, and from here the sperms are supposed to move up to the entire length of uterus. Then after they have to cross half of the fallopian tube, then they will reach here, and then the fertilization will take place. This is normal process. Now if someone is already suffering with the problem of oligospermia, that means less sperms, then what we can do? We can do one thing, we will collect the sample of semen. Now imagine that this is semen. Now this semen is treated and only healthy sperms are taken out of this thing. Then after, these healthy sperms, they will taken into an injection. Treated sperms. And then after, with the help of a pipe, they will directly injected into the uterus. So sperms will be sprayed here, they have to travel less. This process is named as IUI, intrauterine insemination. We are done with one more thing. Now next is IVF. So what is IVF? See, whenever I am saying in vitro for anything, that means I am trying to mimic the biological process in lab. Anything which is mimicked or copied in the laboratory in the same way as it is done by the biology. in the body is named as in vitro. For example, if I'm saying in vitro digestion of food, that means I'm going to use the same enzymes but in the lab to perform the digestion of food in the same way as my stomach is going to digest the food. Now, in vitro fertilization. For this case, only 3 steps you need to remember for prelims level. Number 1 is, first of all, retrieval or female will be injected with ovulation hormones. Now, what is ovulation hormones? These are the hormones, they will induce the ovulation. Now, second thing is, ovulation will now become multiple ovulation. You know very well that in general, a female will produce 1 egg every month, but here she will produce multiple ovum. Now, third thing, doctor will collect the unfertilized egg. Fourth thing is, here is female gamete and here is sperm. Now, in laboratory, fertilization will be performed. Now, as a result of this, you will have a fertilized egg. This fertilized egg, like every fertilized egg, will carry 2n chromosomes, and after some time it will start to divide. And this division will lead into formation of multiple cells, and after this will be implanted to the body. Body means uterus of the mother. And as a result of this, the mother will conceive, and after pregnancy, she will give birth to a baby, and that baby will be named as test tube baby. Now, always remember, whenever you are appearing in exam, they can ask you facts. RPSC is famous and infamous for asking the facts. So, the first test tube baby of world was Louise Brown. Louise Brown. She was born in UK, Louise Brown. And first test tube baby of India, her name was Harsha. Right now she is living in Telangana. She was born in 1986. She was born in 1978, Louise Brown. Now, one more name is there which you need to retain is Kalpana or Durga. She was born in West Bengal. She was born in 1978. She is also claimed to be, sir, I'm Hindi medium student. I can understand English. Good. Okay, so now Kalpana or Durga, she was born in West Bengal. She was born in 1978. She is also claimed to be the first test tube baby of India. So in the options, if there are both the names present, Harsha and Kalpana, then you have to tick Kalpana. But I'm telling you one thing, first certified test tube baby of India is baby Harsha. Durga Agarwal or Kalpana Agarwal was a disputed case. Why it was disputed case? Just listen one line. The doctor who claimed this thing, he was a Bengali doctor, name was Dr. Chattopadhyay or Bandhopadhyay, something like this he committed suicide after this controversy. So this case is not considered as authorized first test tube baby of India. But first test tube baby of India, if they are going to ask you just the name, then remember she was born in 1978. And in case of Rajasthan, if I'm going to talk to you, then I can say thousands of babies are test tube babies. But when we were studying in 11th or 12th, at that time we came to know that there was a girl, her name was Kirti, and she was a magician. She was performing shows all over the India. In her advertisement, I can recall, I have read this thing that she was claiming that she was the first test tube baby of Rajasthan. It was claimed that time. But I'm not going to say that you need to remember that name also. But in case of India, if they are going to ask you, you have to answer like that. So we are done with test tube baby, and we are done with the IUI also. We are done with sperm donation also. Now next is surrogacy or surrogate mother. Now, very easy topic, just a concept and you are done. We are going to hit the bullseye today. Again and again, I'm telling you, after every 10th minute, you are supposed to done with one topic. If you are not doing it, it is not useful to attend this class. So now see, surrogate mother, what is this? There is a lady X. There is a husband of X, Y. And there is other lady, small x. Who is small x? She is sister of X. And who is Y? Y is husband of X. Now just imagine I am Oh, thank you, junior actor. Wow. Now see, now just imagine that I am sitting there and I am in charge of some kind of hospital which is running a facility to help the people who are suffering with the problem named as infertility. And this couple, they have approached me that we are not able to get a baby. So what I will ask that, are you capable to produce OA? She will say yes. Then I will ask him, what about your sperm count? He will say my sperm count is also excellent, but the problem is there is issue with the uterus of lady X. Now I will ask, what is the problem with the uterus of lady X? She will say that I am not able to, according to medical science, I am not able to retain the baby. I am not able to retain the baby for 9 months in my uterus due to some kind of complication. So now X, there is problem with the uterus of X. Uterus of X is compromised. That means there is no problem in conceiving the baby. The problem is she cannot keep the baby for 9 months in her womb. So now what I will do, I will perform IVF. You remember IVF? We have studied it. So I will perform IVF on X and Y, and after IVF I will get a zygote, and zygote will convert it into embryo, and this embryo will be implanted into the womb of X. So this X, who is sister of X, she will be pregnant, and then after she will deliver a baby, or a baby will born out of this, and this baby will baby of X and Y, capital X and Y, but it will take birth from the womb of small x. So in the entire case, small x will be considered as surrogate mother. Now one more thing you need to remember about this is Surrogacy Technology Regulation Bill 2019 was passed by Parliament of India to ensure that surrogacy should not be used for commercial purpose. Always remember this thing. India was converted into the surrogacy hub. Why it was converted into surrogacy hub? The foreigners, they were habitual to come to India. They landed in India, then after they hired a lady and they were taking the advantage of, you can say, poor people. And after 9 or 10 months, they are roaming here and there for 9-10 months. They have, you can say, they have hired a bullet on rental and they are roaming here and there, and after 10 months, they are going with the baby. Now, who is, you can say, exploited here? the lady who is working as surrogate mother. So that's why in regulation bill there are three things which you need to remember. One is government have fixed a criteria, age related criteria. What is the age related criteria? After the age of X years, male and female, they cannot go to have you can say the facilities to acquire the facilities of surrogacy. Second criteria is if any couple is having a living healthy child, I'm using the word healthy child, they cannot go for the surrogacy. And the third thing is surrogate mother should have her own child. And the fourth and last thing is the couple is supposed to pay for the insurance, medicines and post you can say process insurance of the surrogate mother. That's how we have legalized the process of surrogacy, and surrogacy is only possible in India if the doctor is going to certify that in written. The doctor specialist is supposed to certify that this couple needs surrogacy treatment. Then only surrogacy is allowed. So we are done with surrogacy also. Any problem? If no problem, then type Y. I will move to the next topic. If any problem, then please ask me now. Till then, let me drink some water. And I'm telling you, in between, I will tell you very important topics like PCR also, polymerase chain reaction, because entire thing is covered under red biotechnology. Only one Y. What about others? All the juniors, I want attendance. Is IVF harmful more than after 1 or 2 times? First of all, they are not going to ask you this thing in exam. But let me tell you one thing. For IVF, usually they are supposed to try multiple times. And IVF is not harmful. It is a kind of mimicry of the same process in the body of female. Sometimes due to the injections of hormones or sometimes due to the injections of heparin. Heparin is injected into the lady. There can be complications. Otherwise, there are no complications. Okay. All the juniors are present. Okay. One question was there. What is the age criteria for the Surrogacy Act or mentioned in the Surrogacy Act? So the age criteria is under Surrogacy Regulation Act. Under Surrogacy Regulation Act, the lady who is willing to perform the role of surrogate mother should be between the age of, you can say, 35 to 45. 35 to 45. And in the same way, one more thing you need to mention, this act is about that no third party can be involved into the deal of this surrogacy. Many times this topic is discussed in foundation classes in details, because surrogacy, if I am a female and I want to perform the role of surrogate mother, then it is under the article 21. Why it is under article 21? Because it is personal liberty. Recently, in one of the judgments, Supreme Court, the same justice which is right now the Chief Justice of India, Chandrachud, Honorable Chandrachud sir, he said, "My body, my choice." He used this line, quote unquote, "my body, my choice," in one of his judgments. And he said, female is free to do anything with her body. So why you can stop her by means of, you can say, using her body as a surrogacy, you can say, medium. So that's why there are many petitions which are pending in the courts and still the discussions are going on. So all you need to remember is this bill was introduced in 2019 and it was passed in 2021. That's all you need to remember. Okay. So now I'm going to move towards the next thing. Okay, one more thing which was very important and you need to understand is golden rice. Golden rice. Now, you know very well that balanced diet, that's how we are going to cover this entire syllabus. Balanced diet is going to carry at least 1 to 2 questions in your syllabus. Now, first of all, I will give you a brief summary of balanced diet, and then after we will keep on continuing with this thing. So, which diet is balanced diet? So, you need to understand that balanced diet means any diet which is giving you proper energy plus proper nutrition. Now, energy and nutrition, they are not different, but here they are different because energy means you are talking about carbohydrates, plus you are talking about fats, plus you are talking about proteins. And whenever I am talking about nutrition, that means you are talking about the components which are not going to give you energy, but they are necessary. For example, vitamins, for example, minerals. Now, in case of vitamins, whenever you are talking about to deal with vitamins, you need to understand vitamin A. The role of vitamin A is very important and crucial when you are talking about vision. Now, in your eyes, you need to recall your 10th class, 10th standard, chapter number 10 or 11, I think, the colorful world in our surroundings. There is a layer named as retina. Now, in the retina, there are 2 types of cells. One is cones and another one is rods. Now, cones are responsible for color vision, while the rods, they work on a chemical named as rhodopsin. Rhodopsin. Rhodopsin is produced by a chemical reaction with the help of opsin. So whenever I will, for example, right now I am standing in highly, you can say, illuminated area. They have fixed a lot of studio lights here and there, and my eyes are now accommodating, or they have adjusted as per the availability of light. Just imagine what will happen if I will move suddenly from this place into a room which is not having so much lights. So my eyes will take some time, and then after I will be able to look like this. You might have experienced this thing when you are going on a road and some truck or some heavy vehicle have passed from there, and the lights, they focused into your eyes, high beam lights. After that, for some, you can say, a kind of, you can say, milliseconds, you were not able to look anything, and then after your eyes have accommodated. So in between this, what happened? Opsin and rhodopsin. Always remember this thing, opsin and rhodopsin. Opsin is used to look into, you can say, brighter area, and rhodopsin is used when you are looking anything in less light. So your night vision is associated with rhodopsin, and your day vision is associated with opsin, and opsin is continuously capable to convert into rhodopsin, and rhodopsin is capable to convert into opsin. Now both the chemicals, they are precursor of each other, but both of them are dependent of one more chemical which is named as retinol. This retinol is vitamin A. So whenever you will intake less vitamin A, there will be decreased level of retinol in your body, and the decreased level of retinol in your body leads to decreased level of rhodopsin, and decreased level of rhodopsin leads to night blindness, which is which can also be mentioned as if someone is not able to watch anything clearly in less light or in dim light, or someone's rods or the functions of rod cells in the retina is compromised, then it is named as night blindness. So night blindness is directly associated with the deficiency of vitamin A. Now there are many sources of vitamin A, but the source which is most important is golden rice. Now first of all, we will try to understand what is golden rice. Golden rice is GM rice. Again, GM means genetically modified rice. Rice is named as Oryza sativa. Oryza sativa. Now there are two systems. One is PSY gene system, other one is CRTL gene system. These two gene systems are taken from different sources. This CRTL gene system is taken from a bacteria, and PSY gene system is taken from a plant named as daffodil. You know very well, it is you can say it is used for decorative purpose, yellow colors, big size flowers. And this, from daffodil you have taken PSY gene system, from bacteria you have taken CRTL gene system. Now one more thing I want to request you people, and I want to share with you people, you should use this class to understand the concept. And if after that you want to know that what was the name of this enzyme, or what was the name of gene, what was the exact name of bacteria, then you can use your internet resources. Then after you can quickly understand this thing. Now see, by insertion of PSY and CRTL gene, the rice, genome of rice plus PSY plus CRTL. Now this rice will be able to produce a chemical named as beta carotene. Beta carotene. Now you will say, what is beta carotene? Beta carotene, when it will go into your body, this beta carotene will convert into vitamin A. Vitamin A can convert into beta carotene can convert into vitamin A. And when vitamin A is available to your body will produce rhodopsin, and rhodopsin will produce opsin, and vice versa. And as a result of this, you can cure the vitamin A deficiency or the problem of night blindness. Now the things which are supposed to be studied here is, number one, they have produced golden rice 2 also. This is containing higher amounts of beta carotene. One thing. Second thing, so far in India, golden rice is not approved. I am again and again I am sharing with you, the only GM crop which is approved in India for use is GM cotton, BT cotton. No other GM crop is approved by Government of India for the commercial use in India. Now see, in India, golden rice is not approved, but again in Bangladesh, they are using it. USA, they said it's okay to eat golden rice. In Canada, they have approved it, and recently they have approved in Philippines. You know Philippines? Why Philippines is important? Because in Philippines there is Rice Research Institute. And one more thing I want to add here, Rice Research Institute of India is in Cuttack. Cuttack is in Odisha. Now you need to mention in the comments, we are done with one more topic, is there any problem? If no, then type Y, we will move forward. Please be energetic. When I'm asking you to do something, you people are supposed to follow that quickly. You people are the source of my energy. And people who were there present in the classrooms last year, you people are aware of very well, you people are aware very well about one thing. And for me, your response is energy. Achala Gupta, Anshika, I can recall the names. And there are Diksha Yadav. There was one Diksha Sharma also. Now we are going to deal with very important topic. Which topic? This will be quite complicated for the people who are not from science background. But I'm telling you, this will help you to feel that you are done with most of the parts of biotechnology. Now, what is the next thing we are going to done? Hello from Batinda. Wow, good, good, good. Kidda Batinda ch mi penda utthe? IEC Dholpur. Yes, I'm feeling very nervous because exam. See, everyone is supposed to feel nervous. There is nothing wrong in that. Just before the exam, many a times people feel nervous. All you have to realize is take a deep breath and then think, exam to dena hi hai. Why to be scared? Let's go and enjoy the exam. Okay, so let's start about the next thing of biotechnology. We will try to discuss a lot of things about biotechnology. Before that, we need to discuss about what? DNA. DNA and chromosome. So first of all, whenever you are talking about DNA related technologies, you need to understand the packing of DNA in the chromosome. How the DNA is packed within the chromosome. So I think you people are aware of very well aware of one thing, that DNA means what? DNA means, first thing is, it is an example of natural polymer. Second thing, DNA is example of polynucleotide. Now what is this polynucleotide? Poly means many, nucleotide means what? Sugar base, nitrogen base plus sugar plus phosphate, etc., etc. Now see, in case of DNA, on this stage of your preparation, you need to understand 2 or 3 things. Yes, obviously, double helix model, Watson and Crick, adenine, cytosine, triple bond. After cytosine, there will be triple bond. Let me draw the bonds in different color. 1, 2, 3, 1 and 2. People who were thinking to skip this topic can do only this much. And with this much only you can say that yes, we are done with the topic. Adenine, thymine, cytosine, guanine. Now these bonds are hydrogen bonds. One thing. Second thing, from here to here, within the one curl of this, there are approximately 10 pairs of adenine, thymine, cytosine and guanine. This is the basic of DNA you need to understand. Okay, one more thing. DNA means sugar. Which sugar? Pentose sugar. Plus, there are phosphate groups. These phosphate groups and pentose sugar, they are making these arms of DNA. Plus, there are nitrogenous bases. The nitrogenous bases are of 2 categories, purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are cytosine, thymine and uracil. Always remember one thing, uracil is only found in RNA and thymine is only available in DNA. So in case of DNA, there are adenine, guanine, cytosine and thymine. In case of RNA, there are adenine, guanine, cytosine and uracil. Remember this thing, especially the people who are not from science background. Everyone, good evening, hello. People who have joined us late, hello to them. Now see, what type of technology we need to understand from this thing. First of all, we will talk about PCR. And nowadays, the hot topic is RT-PCR. Full form of this thing is reverse transcription polymerase chain reaction. Now many of you can ask that, sir, we have heard one more name, that's real-time polymerase chain reaction. Yes, I agree with you that real-time and reverse transcription, there are two different names, but I can assure you one thing, just stay for 10 minutes with me. With all your ears, you have to listen to me. With all your senses, you have to focus here. If you will do this, PCR technology will never bother you in life. This is promise of Dharmendra was to you. I can give you guarantee. All the juniors who are my students or who are my students for this session, everyone, remember my promise. I'm giving you words for you people. Today onwards, the most, you can say, easy topic of the world will be PCR. And you will be very happy that yes, now we can understand what is PCR and what is reverse transcription. So now with all your ears, I want you people to understand this thing, that DNA means there is a coiled structure, and within this coil there are adenine, thymine. They are connected with each other with the help of double bonds, and there is cytosine and guanine. They are attached with each other with the help of triple bond. These triple bonds are, yes, hydrogen bonds. Double bonds are also hydrogen bonds. On this side of DNA, there is, on this side there is there are two you can say arms. These arms are made up of pentose sugar, deoxyribose sugar, and then after there are phosphate groups. So that's how DNA is made up of. Now, whenever you are talking about RT-PCR, you need to understand one concept. What is that concept? The concept of central dogma, or the principle of protein synthesis, or the mechanism of protein synthesis. First of all, they will ask you, this was prompted by whom? So Watson and Crick, there are two scientists. Whenever you are talking about structure of DNA, here in case of central dogma, you have to talk about only and only Crick. This was suggested by Francis Crick, not the James Watson. James Watson was working with Francis Crick when both of them, they have suggested the double helix model of DNA. Here you have to remember only Francis Crick. Now what is central dogma? Central dogma is saying that protein synthesis is taking place in 3 steps. Number 1 is replication. Number 2 is transcription. And third one is translation. Now, what is transcription, translation and translation, or transcription and translation? See, this is a cell. Within cell, there is nucleus. Within nucleus, there is nuclear material, or I can say DNA is lying here. DNA means double helical structure. Now, this DNA is carrying genetic information of what type of protein will be formed there. Now, this genetic information is lying within DNA. So, first of all, in the process of replication, this DNA will replicate to make a copy of DNA. Copy of DNA. So, first of all, DNA will form a copy of DNA. As I told you, this is bit complicated in starting, but after 5-10 minutes, this will be the most easy topic for you people. And most of you are unaware of the, you can say, concepts behind the PCR technology. And you know very well that this is very important nowadays, especially after the COVID. It is very important. For example, let me give you one example why this is so important. I am drafting a question here. PCR, especially I am writing RT-PCR, is used detection of A, B, C, D. Now, number 1, number 2, number 3. TB, cancer, AIDS, fourth is COVID-19. Now options, as you know, in civils exam, they are going to ask the questions like only 1 and 2, only 1 and 4, only 3, and then all 1, 2, 3 and 4. Answer it. And there are many students, around 50 students are there. I want most of you to comment here. If my class is useful for you, first answer this. Then I will start it. And I want everyone to answer this honestly. All you are supposed to do is answer, or type A, B, C, D, whatever is your answer. Just type A, B, C, D, whatever. As you can see, most of you are answering B. Why most of you are answering B? Because you people are carrying a mindset that PCR means viral infections. But the real answer is all of them. Yes. It is used as a marker to find out the cancer cells also. That's why I said. That's why I asked you people to answer this thing. It is not a, you can say, easy topic to understand, but this is the basic concept of entire biotechnology, especially when you are talking about red biotechnology. Now see, whenever you are talking about PCR, you need to understand polymerase chain reaction. First of all, you are going to amplify the DNA segment. Why you will amplify the DNA segment? You will amplify it to find out whether the DNA is carrying the suspected segments of the codons or not. So thank me later, you can thank me later. I have saved you people from making this mistake in the exam. Now see, central dogma or protein synthesis mechanism. So first is replication. Replication means DNA will form DNA. Second is transcription. Now what happened? See, you people have studied about the structure of cell. Now in the structure of cell, this is nucleus. Now within nucleus, one cell organelle is formed that is named as ribosome. Always remember, ribosomes, they are formed in nucleus, but they left nucleus. So here are the ribosomes. They were formed here, but now they are here. So I am erasing this link. Now these ribosomes are named as protein factories. Now, which type of protein should be formed? That information is lying here in DNA. Let me make it red again so that you people should not get confused that, sir, you have made it red earlier and now how it can be black. And in first, you can say process, DNA has prepared a copy of DNA. Now, there are pores in the nuclear membrane. There are pores. And from these pores, DNA will not go. Who can cross? From here, RNA can cross. So now this DNA will form one type of RNA. Always remember, RNA is single stranded, it is not double stranded. So that's why I am making RNA like this, and this RNA is carrying the information which was lying in the DNA. That's why it is named as mRNA. This is named as mRNA. This was DNA, copy of DNA. Now in the process of transcription, DNA has formed mRNA. So now one mRNA is formed, and that mRNA is coming out of nucleus. This is mRNA. Now what will happen? This mRNA is carrying the information. The information of mRNA will be, you can say, studied by the ribosome. And the ribosome will arrange the amino acids as per the information provided in mRNA. So as a result of this, the information is translated. Translation will take place, and as a result of this, protein will be formed. This is the basic of the, you can say, protein synthesis. This was, I can remember, part of 9th standard book when I was in 9th standard. We have studied this thing in 9th class, and I think I am having that book also. Now see, that's how the protein is synthesized. Here you have to remember one thing: whenever we are forming RNA is formed from DNA, this process is named as transcription. Now RNA from DNA, formation of RNA from DNA is named transcription. So what will be reverse transcription? Reverse transcription will be formation of DNA formation from RNA. So here, in case of reverse transcription, first of all, reverse transcription, RT, we have to understand. It will be converted into DNA. That's why it is named as reverse transcription. Now, we will try to understand what is PCR, polymerase chain reaction. This is one segment, RT, reverse transcription. Reverse transcription is formation of DNA from RNA. Now the second type, second part. Second part is, what is PCR? Polymerase chain reaction. Polymerase chain reaction is technique or process to amplify the DNA. We have already obtained DNA, now we are going to amplify it. Now the amplification of DNA is performed in a special machine. Now listen carefully. Amplification of DNA is performed in a special machine. This machine is named as, so first requirement is machine. This machine is named as thermocycler machine, or also named as PCR machine. Now why it is named as thermocycler machine? Because with the help of variation of temperature, different type of temperature is maintained over the machine during the entire process, and that's how the machine works. Now what are the steps which are involved in PCR? So now, many times they will ask you to arrange these steps. So the first step is, DNA is like this. Actually it is like this. But here we are drawing it like this. DNA is like this, and there are adenine, guanine, cytosine, thymine like this. Now, first process is denaturation. Denaturation means with the help of temperature, and temperature is maintained around 95 degrees Celsius. Both the ends are, you can say, uncoiled. लाइव कम होने का रीजन हिंदी मीडियम में ना होना है, डजंट मैटर फॉर मी. लाइव तो आई डोंट नो हाउ मेनी पीपल आर देयर. 54 are watching. Okay. Now see, so first is denaturation. Is it performed around 95 degrees Celsius? So first of all, they will ask you that how the PCR is performed. You have to say first step is denaturation. And what is denaturation? In PCR cycler or in thermocycler machine, on the temperature 95 degrees Celsius, we will separate out both the you can say ends of DNA. Now second is annealing. Now, what is annihilation or annealing? So, in case of annealing, we need the DNA to start to prepare the complementary part. So, now we will add a special segment which is named as primer. Primers are added, and with the help of primers, one primer will be added here. With the help of primers, the process will start. Process of what? The completion of this coil. So the third step will be the completion of this thing. That is named as extension. So this half part of DNA will try to complete it. And with the help of primer, the process is initiated. And that's how from one DNA, you have prepared two copies of DNA. You have prepared two copies of DNA. That's why this process is named as amplification of DNA. Polymerase chain reaction. Now, within few minutes, you have doubled the copy of DNA. If you will run this thing again and again, there you will get a lot of copies. For example, after 20 to 30 minutes, you will have approximately 10 lakh copies of DNA. 1 million copies of DNA you will get. So there was BT mustard in news to be adopted in India for use. Is it relevant? See, don't study anything which is carrying the scope of, you can say, discussion. Don't study anything. Because right now, even I am going in a, you can say, shortcut ways. So you need to understand only three things. Number one, in case of polymerase chain reaction, our purpose is to enhance the DNA. How we can enhance or how we can multiply the DNA copies. So first of all, we will take the sample, and in thermocycler, we will maintain the temperature of approximately 95 degrees Celsius. On this temperature, with the help of many enzymes and chemicals, what we will do, we will introduce temperature, and as a result of this, there will be separation of both the sides of DNA. Now this is named as denaturation. After denaturation, with the help of primers, we will perform the second step, that is annealing. And then after third, third step is, ये क्या discussion चल रहा है अलग ही, मैं तो एक शिवलिंग लाई बस. Okay, now see, after third step, the DNA, the primer will initiate the formation of complementary part, and as a result of this, the DNA will start to amplify itself. This process is named as DNA amplification. Now see, we will talk about use of PCR. Now, why we will amplify the copies of DNA? Now, in machine, we are amplifying the copy of DNA. As a result of this, we will get multiple DNAs. Now, it will be easy. It will be easy for us to detect it now. So, for example, now we are going on the pure, you can say, application part of this thing. Just imagine Achala. I'm taking this name from one of my, you can say, comments, and she is my student also. So Achala, she want to go for the testing of COVID-19. So first of all, COVID-19 is actually happening because of a virus, which is RNA virus. And you know very well that this virus is containing RNA. There are glycoproteins, and these proteins are always shown like this because they are named as spike proteins. They help the virus to attach with others. Now see, she is going through the COVID-19 testing. First of all, sample will be collected. From where the sample will be collected? From the nasopharynx. Just imagine this is the swab, cotton swab. On this, from the nasal route, they have taken the sample. Now, this sample is carrying RNA of virus. Now, second step is preservation of sample. Now, to preserve it, the sample will be transferred into a tube, and this tube is sealed like this, and there will be a liquid in the tube. This liquid in the tube is mentioned as virus transportation medium. Now in this they will put this swab, and then after they will seal it. Now the third step. Third step is in laboratory. They will separate out or extract out RNA from the sample. Now fourth will be ये बीच में अलग ही स्टोरी चल रही है, जय महाकाल एंड RS, इनकी लव स्टोरी चल रही है यहाँ पे. दिल ही नहीं है क्या है भाई? यहाँ RNA इन बच्चों से पूछ के देखो जिनका एग्जाम है. RNA DNA ने दिमाग खराब कर रखा है इनका. मतलब यहाँ पे मैं कह रहा हूं कि सबसे मुश्किल टॉपिक चल रहा है, वो कह रहा है दिल ही नहीं है आपके पास, शिवलिंग लाए हो क्या, ये है वो है. व्हाट इज दिस? आफ्टर 6 days एग्जाम है इन बच्चों का. एटलीस्ट इनको पढ़ने दो, नंबर ले लो एक दूसरे का, तुम घर पे करो जो डिस्कशन तुम्हें करना है यार. व्हाट्सएप है, टेलीग्राम है, इतने सारे मीडियम्स हैं, घर पे करो ना पर्सनल टॉक्स. हमारा ही प्लेटफॉर्म मिला था तुमको ये यहाँ पे 1947 लव स्टोरी चलाने के लिए, या उस जमाने में जी रहे हो कि पत्थर में लेटर बांध के तुम फेंकोगे, फिर वो फेंकेगा जवाब उसका. इनको ब्लॉक करके डिलीट विलीट करो सर. चलो हाँ जी, तो लैब में आके दे विल सेपरेट आउट द सैंपल. व्हाट दे विल डू नाउ? फर्स्ट ऑफ ऑल दे विल डू RT. नाउ व्हाट इज RT? रिवर्स ट्रांसक्रिप्शन. reverse transcription. Reverse transcription means the RNA will be converted into DNA. Then after fifth step will be, now they are carrying DNA. Now what they will do in fifth step? In fifth step, they will run PCR. PCR means again three cycles: denaturation, then after annealing, and third is extension. And again these cycles will be, you can say, running in the cycler. After X amount of cycles, they will run the sixth step, that is test the results. Now after results, if after like 35 or 40 cycles, now just imagine Achala is the name of patient जिसका sample test हो रहा है, and in case of her you can say sample, after 40 cycles, I'm not sure that I am able to detect the DNA. That means she is COVID negative. And after 20 cycles, if I am able to detect the DNA, that means she is carrying very high virus load. That's how this process is done. Now in this process, one more thing I want to tell you. Whenever you are amplifying the DNA, it is not only used DNA amplification. It is not only used for diagnosis of you can say disease also. It is also used for DNA fingerprinting also. In case of DNA fingerprinting, they run the same thing. Actually, it is named as DNA profile testing. It is not named as DNA fingerprinting. It is named as DNA profiling. So in case of DNA profiling, they will also run the same thing. This is the sample of DNA which is recovered from the case. This DNA sample is going through the same process: denaturation, then after annealing, and then after extension. That's how we will, first of all, we will amplify the sample. Amplification of sample. After amplification of sample, you will have multiple DNAs. Not one, not two, in millions you will have copies of DNA. These copies then examined with reference to the DNA sample. That's why DNA profiling is a reliable test in case of criminal cases like rape, murder, identification of dead body, or the cases related to paternity testing. Paternity testing means who is the father of the baby, etc., etc. It is also used in research, you can say, purpose. In India, always remember DNA laboratory is in Hyderabad. In Rajasthan, DNA testing laboratory is in SMS. And SMS hospitals laboratory is carrying a record. What is the record? They have filed speediest results in case of criminal case, which was a case of that case was famous with the title Jivanu in Rajasthan. There was some psychopath who was infamous for his heinous acts against the girls, especially girl child. So he was jailed after this thing. And in India, after 1987, DNA profiling is considered as an evidence. Got it? So we are done with one more topic. Once again, I'm going to revise it for you people. Whenever you are dealing with PCR, polymerase chain reaction, first of all, polymerase chain reaction. Polymerase chain reaction means you are carrying a copy of DNA, you want to amplify it first. To amplify it first you will open the coil of DNA. When you open the coil of DNA, there is A, here is T, here is C, here is G. So you have opened it. Now this chain and this chain, they are half. Now they will try to complete them. Now to complete them, we will initiate a process that is initiated with the help of primers. Now when primers will initiate the process, the entire DNA will complete itself. So that's how copies of DNA are prepared. that is named as extension. So denaturation, annealing, and extension. And again, if you run these cycles, you can say that I am completing the process of polymerase chain reaction. So now type Y if you want me to proceed to the next topic. I'm waiting for your Y. Okay, good. Singodhya is also my student. I can recall her name also. Anshika. Okay. Now see, next topic is very important. Next topic is recombinant DNA technology. Now see, it is almost 2.5 hours and we will be done with 90% of biotech after this topic. So in 3 hours, we will be done with one entire unit. Got it? Now see, recombinant DNA technology. First of all, you need to understand the use of recombinant DNA technology. For example, I think you people are aware of one thing, that chromosomes, they are made up of chromatins, and chromatins, they are made up of DNA, and DNA is carrying genes. That's why genetics and biotech, they go always hand in hand. Now again I am telling you, chromosomes, they are carrying chromatins, chromatins are carrying DNA, and DNA is carrying genes, and genes are responsible for characters or qualities. Now see, first of all, in case of recombinant DNA, we have to identify the target gene. Now what is target gene? Target gene means I not समझ आवेंगे in English. No problem. Okay. Now see, identifying the target gene. Target gene means, just imagine, here is a segment of DNA. Now just imagine this red part is the part of gene responsible for production of insulin. This red part is the reason because of which you can get insulin. Now second thing, so this is identifying. First of all, we are going to identify that which part is our target gene. Target gene, or we can say gene of interest. Interest means in which we are interested. Now what we will do? We will isolate the gene of interest. Now how we will isolate the gene of interest? For this we will use molecular scissors. Now what is molecular scissor? Molecular scissor is an enzyme named as restriction endonuclease enzyme. There are many type of enzymes under the heading restriction endonuclease enzymes. right now you have to keep one thing in your head. The first thing I want you people to write down is restriction endonuclease is the enzyme which is capable to cut the DNA from desired place. From wherever you want, you can cut the DNA. It will cut the DNA for you. So this was your DNA. In your DNA, this was your gene of interest, and with the application of restriction endonuclease enzyme, you have taken out this much segment of DNA. Now this segment of DNA is carrying gene of interest. Now listen carefully. Third thing is we will go for the vector. Now what is vector? Vector can be a plasmid. What is plasmid? I will tell you. Vector can be yeast cell, chromosome. Vector can be mammalian cell, artificial chromosome, or vector can be a bacteriophage. Out of them, the only thing which is useful for you is plasmid. Why only plasmid is useful? Because even in BSc books, plasmid is mentioned. All others are mentioned either in medical books or in biotechnology books. So you need to understand vector means plasmid. Now what is plasmid? In eukaryotic cells, especially in bacterial cell, this is the genetic material. Other than this, there is a circular DNA. This circular DNA is named as plasmid. This is bacterial cell. And this is genetic material of bacteria. Now what we will do, we will take out the plasmid. We have taken out the plasmid. So what is plasmid? Plasmid is circular DNA. Circular, I should write down extra DNA. Why extra DNA? Because it is other than the genetic material. Now what I will do, I will apply the same restriction endonuclease enzyme on it. When I will apply restriction endonuclease enzyme on it, the restriction endonuclease enzyme will cut it like this. And on the other hand, if you will see, I have already taken out the gene of interest, the gene which is capable to create insulin, the gene which is capable to produce insulin. Now what I will do, so this is, I have taken the vector. Now what I will do, I will take the gene of interest. Remember this segment is gene of interest. Then after, I will add that gene of interest into this circular DNA. So as a result of this, okay, to add them, I need one more enzyme. That is named as ligase enzyme system. Remember students, this question has been asked in RAS 2013 prelims. Again they have asked in 2016 prelims. Which enzyme is named as molecular tailor or DNA tailor, or which is used for joining the DNA? Answer should be ligase. And which enzyme is used for cutting the DNA from a targeted place? The answer should be restriction endonuclease. That's why restriction endonuclease is mentioned as molecular scissors, and ligase enzyme is mentioned as something which can join the DNA parts. Now with the help of ligase enzyme, what will happen? We will be able to prepare something like this. This is plasmid of bacteria, and within plasmid bacteria you have inserted gene of interest. Now next step, we will insert this DNA. Now this DNA is named as recombinant DNA. Recombinant DNA. Now this recombinant DNA will inserted into host cell. Insertion to host cell. Now this host cell most of the time is E. coli bacteria. Even bacillus bacteria can be used, but most of the time it is E. coli bacteria. So in Uttarakhand PCS prelims, they have asked last time, they have asked the question, which bacteria is used in biotechnological applications, specifically on molecular levels. So your answer should be E. coli, Escherichia coli. Now Escherichia coli is something like this. Now in Escherichia coli, you have inserted this plasmid. And this plasmid is carrying this much gene of interest. Rest of the things are of E. coli. Now, as per the nature, E. coli will reproduce. And as a result of this, multiplication of recombinant DNA will take place. And you will have copies of this DNA. From 1, you will get 2. From 2, you will get 4, 8, 16, 32. Like that, millions of copies you will get. Now, because this gene of interest is capable to produce insulin, so as a result, listen carefully, as a result, this bacterial product will carry insulin. So that's how insulin is produced in the laboratory with the help of bacteria. Hormones are produced, enzymes are produced. For example, injections of HGH are available. HGH is what? Human growth hormone. Human growth hormone is also named as pituitary. Now, growth hormone is useful to help the candidates who are suffering with the deficiency of human growth hormone. So far, the only source of growth hormone was to kill someone and take out the pituitary gland of that person. But now, without interfering with the functions of pituitary gland, you can get out this hormone. How you can get out this hormone? All you have to do is you have to take out the gene of interest, the gene which is responsible for formation of this hormone. Then after you will insert this gene into the plasmid, you will prepare a recombinant DNA, and then after that recombinant DNA is inserted into a host. That host will usually be E. coli. And then after multiplication, it will start to produce growth hormone for you. You get something out of it or not? Please comment. Because from my side, we are done with one more topic. Please comment. And I will be back in 2-3 minutes. Kindly excuse me. So, what are the comments? Okay, done. Okay. So, many of you are done. Very confusing. Yes, it is quite confusing. Priyanka ma'am. Okay, Priyanka Srivastava. Okay, ma'am. Good evening. So, we are done with most of the things of this biotechnology. So now the only thing which I think is left is CRISPR-Cas9. Yes, we are going to talk about CRISPR-Cas9. Don't worry. We will discuss about CRISPR-Cas9 right now. Anshika Agarwal. Yes. So now what is CRISPR-Cas9? We'll try to understand it. And you know very well that CRISPR-Cas9 has been asked in IAS also. Not only once, twice or thrice they have asked the questions from this thing, CRISPR-Cas9. So first of all, what is CRISPR-Cas9? The entire thing is dependent of one protein that is named as Cas9. CRISPR is the name of process, and what is Cas9? First of all, we will try to understand it. Just imagine this is a cell of bacteria. This bacterial cell is attacked by a virus, and this virus is containing DNA. The DNA of virus, I'm drawing it in red color. So in next stage, you know very well that virus is the organism which is considered as a linkage between live and dead. Why it is considered as a linkage between live and dead? Because this organism is not able to reproduce itself. So how they multiply their number? So the virus will inject its DNA into this bacterial cell. And then after this DNA of virus will hijack the cell of bacteria, and by utilizing the cell organelles of bacteria, it will multiply. It will prepare new viruses, and then they will start to infect the other cells also. Now see, in defense to this, bacterial cell will try to cut the viral DNA. Now, this was the point of interest for the scientists. Bacterial cell will cut the DNA of virus to defend itself. Now scientists, they will try to find out which mechanism is responsible that bacterial cell is capable to cut down the DNA of virus. The DNA which is trying to infect a cell, and cell is cutting it down. So far we were aware of only one thing, that restriction endonuclease enzyme system is capable to cut the DNA. Now who is this chopper? When they tried to find out, they came to know that it is all happening because of a protein named as Cas9. So now what is happening? Cas9 is a protein which will stick or which will attach to DNA and then after cut it. So just imagine this is DNA, and I want to cut this DNA from here. So Cas9 is capable to cut it, but how I will ensure that it should cut it from here? So for this thing, in case of biotechnological application, this is the phenomena of nature which is happening in nature. Now in the biotech lab, what I will do, first of all, I will prepare a tool named as guide RNA. Now what is guide RNA? सर आपके नोबल का नाम, भाई पहली बात तो नोबल नहीं, नोवेल. नॉवेल लिख रहा हूं मैं, है ना, और नॉवेल का नाम है पिंक कैक्टस, और जब आ जाएगा थोड़े दिन में आने वाला है, मैं खुद ही बता दूंगा, यहीं पे आके बता दूंगा दोस्त, है ना, यहीं पे आके बता दूंगा। नोबेल, नोबेल आपको पता है नोबेल प्राइज है, मेरे को लगा मैंने भाई कौन सा नोबेल आ रहा है मेरा, दैट इज नोवेल, उपन्यास। चलो देखो, ठीक है, तो गाइड आरएनए इज अ टूल विच विल गाइड, सो दिस विल guide the Cas9 to attach. So now, to ensure that on X point Cas9 will attach and it will cut it from there, first of all in lab we will prepare guide RNA. With guide RNA, when you will send the second tool, that is Cas9. So Cas9, you know very well, it is DNA cutter. So guide RNA will guide it and Cas9 will cut it from wherever you want to cut. So it is amazing tool. The future of super babies is here. Super babies means, for example, I am having several characters. You people are having several characters. All the characters are actually because of some segment of genome. And all the genes are lying within the DNA. Now, if you want to take out some of the portion, any of the portion of my DNA, then the problem was how to cut it. Now you can do it. How you can do it? You can do it by means of just two things. One, prepare guide RNA. Let's prepare a guide RNA which will attach there. Why the RNA will go there and attach there? The answer is A and T. C and G, A and T, C and G. Wherever you want to attach, you have to prepare the complementary base pair. And when it will attach with it, the guide RNA will tell the Cas9 from here you have to cut it. From here, when you will cut it from here, you will get this much segment of DNA. As a result of this, you can prepare anything. Anything means you can take out the part of DNA which is responsible for aging. You can take out the part of DNA which is responsible for, you can say, any disease. So that's why CRISPR-Cas9 is example of one of the most advanced technologies of DNA repairing. But this will be considered as recombination therapy or gene therapy. Answer in the comments. Harsh and Aditi. Oh wow. Thank you. God bless you both. My question is, CRISPR-Cas9 is example of recombinant DNA therapy or it is an example of gene therapy? Yes, it is an example of gene therapy. Absolutely correct. Now, only thing I want you people to add on here is, for this exam or some other exam, wherever you want to, got it? In 2022, Government of India have approved one plan. What is that plan? Listen carefully. There is a disease named as sickle cell anemia. The cure or the treatment of sickle cell anemia with the help of CRISPR-Cas9. CRISPR-Cas9 technology is under process. Trials are going on and a 5-year program 2022 to 2027 is going on under the supervision of prominent doctors of the country. हिंदी मीडियम की क्लास कब होगी? आफ्टर 2-3 days. Got it? So this is Cas9, and hopefully you people are aware of full form of this thing. I don't think they will ask this question, but even if they are going to ask, you have to remember C for clustered, R for regularly, I for interspaced, S for shorts, P for palindromic, R for repeats. Clustered, regularly, interspaced, palindromic repeats is CRISPR. And Cas9 is the protein which is produced by the bacterial cell as a defense mechanism. Now, if you people will type 5y in 1 minute, then I will finish one entire topic in next 10 minutes. And type only if you are getting it. One Y is there. Okay, that means you people are not sleeping. I am happy. Okay, so now see, there was a term named sickle cell anemia. Sickle cell anemia. So why I said that I will finish entire topic. Now you know very well that general science is one of the integral parts of your syllabus. In general science there are two topics of biology which are carrying at least 3 or 4 question weightage. One of them is blood. Whenever you are talking about blood, we will finish it in single shot. How we will finish it in single shot? Just imagine what is blood. So blood is a tissue. Blood is a tissue, connective tissue. Now, from blood, we will take 1 or 2 topics of biotech also, but within 10 minutes, we will finish it. Now, within blood, there is approximately 45% of blood is RBC plus WBC plus platelets. Then remaining 54-55% is blood plasma. Always remember one thing, that if you will centrifuge the blood, you know very well centrifugation. Whenever anything is in rotatory motion, 2 forces are working on it. One is centrifugal force, other one is centripetal force. Centrifugation means going away from the center. So, as the effect of this force, you can separate out the components of blood. How they will be separated? In the lower portion, you will get RBCs. Now, because they are red, so this portion will be red. After that, there will be a yellow layer. This yellow layer is WBC plus platelets. And above all, you will get so-called plasma. Plasma is approximately 55%. Now, whenever you are talking about plasma, you have to understand one thing. What is that? Plasma therapy. Now, what is plasma therapy? What is plasma therapy? To understand this, you need to understand the recipe to prepare plasma. Now, to prepare plasma, you need to take approximately 92% of water. Plus, you have to add 3 types of proteins. One is globulins, one is albumins, and one is fibrinogens. With this, you have to add a lot of ions. For example, magnesium, calcium, chloride, sodium, etc. Plus, you have to add hormones. You have to add enzymes, and you are supposed to add blood clotting factors. When you will add this much of stuff, you will say this is plasma. So obviously, when they are going to ask you what are the components of plasma, you have to remember the entire thing. But out of this, the most important is role of these 3 types of proteins. Always remember globulins. Globulins are actually going to produce immunoglobulins. Immunoglobulins, IG. You know very well, IgGs are your antibodies. IgGs are of 5 types: IgG, IgA, IgM, IgE, and IgD. Now, first question for you people is, you need to answer right now, which Ig type of antibody is found in breast milk. The first breast milk is named as colostrum, and it is advised that it should be, you can say, available for every kid. Now, my question for you is, which Ig is found in breast milk? First question. Second question is, which Ig or which immunoglobulin is capable to cross the placenta? You need to understand and you need to find these two concepts. If you don't know, then Google it right now, because you need to understand what you will answer if tomorrow they will ask you that which antibody is provided to the kid when he is in womb of mother. Or they can ask you that which antibody is the first antibody which is provided to the kid when the kid is intaking the milk of mother's breast. Two things. And third thing, from here you have to understand the concept of passive immunity. The meaning of passive immunity is, if I am getting immunity by indirect action of someone. Indirect action means the action which is not prepared from my side of immune system. That is passive type of immunity. So don't you think if the immunity which you are getting from the mother's milk is passive immunity? One. Second, passive, but it is natural immunity. The immunity which you are getting from the placenta of your mother, that is also passive, but that is also natural immunity. So that's how you have to understand a lot of things. I'm waiting for your comments. Okay, now see, so there are 5 types of antibodies. All of them are associated with globulin proteins. Now, albumin. Now, albumin, whenever you are talking about albumin, you have to say albumin is responsible for osmosis balance. Now, what is osmolality or what is osmosis? Now, see, you have to understand there are certain semi-permeable membranes. Semi-permeable means the membrane which will allow something to pass through and which will not allow everything to pass through is named as semi-permeable. It is named as permeable when it will allow everything to go through. Now, see, on one side, if there is a solution of higher concentration, and on the other side there is solution of lower concentration, then what will happen? The particles of solute particles will move from higher concentration to lower concentration, and particles of solvent, they will move from lower concentration to higher concentration. The movement of solute particles from higher concentration to lower concentration, and movement of solvent particles from lower concentration to higher concentration is the process, is the phenomena which is named as osmosis. And just reverse to this is happening in the RO of your kitchen, which is reverse osmosis. That means in RO, the solvent particles, remember this thing, solvent particles in RO, solvent particles will move from, yes, higher concentration to lower concentration. And in case of reverse osmosis, solute particles will move from lower concentration to higher concentration. That's why that is reverse osmosis. That's why that process needs a lot of pressure. That's why there are special type of membranes used there. Now see, in case of cell, every cell is semi-permeable. Why? Because there is cell membrane. Remember that cell membrane is made up of proteins plus lipids, and these proteins are glycoproteins also. That's why cell membrane is semi-permeable. Now, across this cell membrane, solute and solvent particles keep on going from one side to other side, one side to other side. RBC is also a cell. RBC is also a cell, and this is cell membrane of RBC. Always remember the shape of RBC, shape of U. is a song dedicated to RBC. The cell membrane of RBC is also semi-permeable. That's why whenever you are going to inject any medicine into the body of anyone, the concentration of that component is always considered in a scientific way. For example, if you'll go to any hospital and if you will see any patient who is injected with dextrose, it will be written there 0.9% or something like that. That percentage is significant by means of osmosis. So which protein is capable to maintain the osmotic balance? That is albumin. And next protein is fibrinogen. Fibrinogen is responsible for blood clotting. Whenever you will have any type of, you can say, injury, you will you can say the blood loss will stop within 3 to 5 minutes. That is because of fibrinogen. Now next thing is, the shape of RBC is always biconcave. Again, I'm saying shape of RBC is okay. How many RBCs are there? How many WBCs are there? And how many platelets are there? Always remember, number of RBC is approximately 45 to 55 lakhs per ml. Number of WBC is 5500 to 10,000 per ml. Number of platelets is approximately 1.5 lakh to 3 lakh per ml. And one more thing you have to add on here. In case of RBC, many times they can use the term erythrocytes. In case of WBC, they can use the term leukocytes. And in case of platelets, they can use the term thrombocytes. Now, what you will say if there is a question, what is erythrocytopenia? What you will say if there is a question, what is leukocytopenia? And what you will say if there is a question, what is thrombocytopenia? Anywhere if the cell name is carrying a suffix named penia, that means kami hai uska. So if any patient who is suffering from dengue, what is the virus of dengue? Always remember arbovirus, arboflavivirus. Who is the vector of dengue? Female Aedes. Now, whenever any patient is suffering from dengue, he will be a patient of thrombocytopenia. Whenever any patient is patient of malaria, he will be a patient of erythrocytopenia. Why? Because the dengue virus is famous and infamous for killing of the thrombocytes, while the target of plasmodium is always RBC. That's why it will cause erythrocytopenia. On the other hand, in case of GK, you have to remember one more thing. What's that? That thing is, whenever any cell name is suffixed with his, for example, erythrocytosis, leukocytosis, thrombocytosis, that means increased number of cells. So decreased is penia. Got it? Now see, this is the normal range of RBC, WBC and platelets. Remember one more thing, in female this is slightly less. Now RBC is biconcave in shape in healthy human. What will happen if someone is suffering with sickle cell anemia? If anyone is suffering with sickle cell anemia, the shape of RBC in the patient will be like this. This is the shape of RBC. Because it looks like a sickle, that's why it is named as sickle cell anemia. Now see, whenever you are talking about sickle cell anemia, always remember sickle cell anemia is a genetic disorder. It is a genetic disease. Number one. Number two, shape of RBC is actually deformed because of deformed hemoglobin. Now, where is hemoglobin? Hemoglobin lives here in the RBC. RBC is the house of hemoglobin. That's why if any patient is anemic or anyone is patient of anemia, go to the portal of Anemia Mukt Bharat. www.anemia-mukt-bharat. If you will open the portal, you will came to know about the definition of anemia, and official definition of anemia is anyone who is having deficiency of RBC or hemoglobin or both is considered as anemic patient. And as per NFHS, National Family and Health Survey number 5, which is the most recent official reference for any type of health related issues. As per NFHS, the situation is really to worry about, because even in the male demographic dividend, boys from 10 to 19 years, 31% boys are suffering from anemia. More than 50% female who are pregnant are suffering from anemia. More than 50% female who are lactating female. Lactating means they are breastfeeding their babies. They are having anemia. And more than 50% girls who are adolescent, they are suffering from anemia. And more thing to worry about is 57% kids who are from the age group 6 months to 59 months, they are suffering from anemia. Because of all this worrisome situation, Government of India decided to launch a mission. That's why in 2018, we have launched the world's largest, world's biggest program to fight against anemia. The name of that program is Anemia Mukt Bharat. So they can ask you, Anemia Mukt Bharat was launched in 2018. Now one more thing, Anemia Mukt Bharat is operated with a strategy 6 into 6 into 6. Now what is 6 into 6 into 6? First 6 is for 6 target groups. 6 target groups are selected to conquer anemia issue. The first target group is the kids from 6 months to 59 months. Second target group is kids from the age of 5 to 10. Third target group is 10 to 19, boys and girls. Then after fourth target group, female from 20 to 24. Remember, boys only up to 19. After that, only female is target. 20 to 24 is considered as women from reproductive age. Then after, pregnant and lactation, pregnant lady, and then after the sixth group is lactating mothers. Mothers which are breastfeeding their kids up to the age of 6 months. So that's how they have decided 6 targets. Second 6 is for 6 type of interventions. 6 type of interventions. With the help of these interventions, we will be able to complete the aim or target of Anemia Mukt Bharat. So out of this, very interesting thing is, first, yes, we are aware of first thing, that first intervention is we will provide folic acid and iron supplement. Always remember, folic acid is vitamin B9. And folic acid is useful for the absorption of iron. So whenever there will be iron supplement provided to any of the patient, it will always be provided with vitamin B9. And vitamin B9, always remember, vitamin B9 is named as folic acid. So that's why if there will be a question that which vitamin is responsible for anemia, deficiency of which vitamin is responsible for anemia, your answer should be vitamin B9. But there is one technical term, pernicious anemia. So if they will ask you which vitamin deficiency is responsible for pernicious anemia, as soon as you will see the name pernicious, you should feel like, yes, I am enlightened. This is vitamin B12. Vitamin B12 is named as cyanocobalamin. You have to mug up the names of vitamins. Doesn't matter that you have not studied yet. Today is your first day, tomorrow, day after tomorrow, every day if you will remember one name or two names, you will be done with names of vitamins. Whenever you are talking about vitamin, all you need to remember is close your eyes and you have to remember vitamin A, vitamin D, vitamin E, vitamin K. These vitamins are fat soluble vitamins. While vitamin B and vitamin C, these are water soluble vitamins. Whenever you are talking about vitamin A, the name of vitamin E is retinol. Retina is in my eyes. That's why it is associated with all the defects of eyes. For example, we have studied in biotechnology golden rice. Why? Because in India, a lot of people are suffering with night blindness. deficiency of vitamin A or retinol. Now, whenever you are talking about the resources or the sources of vitamin A, you should remember one thing: fish oil, cod liver oil, all the dairy products. Why? Because this is fat soluble. And all these products are carrying fat. Egg is a good source of vitamin A. And with this, don't forget the vaccination program of India. In vaccination program of India, doses of vitamin A, oral doses of vitamin A are included. First dose is given at the age of 9 months. 1 ml dose equivalent to 1 lakh unit, international units. Vitamins are always measured in international units. Next time when you will see the bottle of vitamin, you can read on it. It is written like this much lakh IU. IU is international units. So first dose of vitamin A at the age of 9 months. Second dose of vitamin A at the age of 18 months, 2 ml. And then after every 6th month, 1 dose of vitamin A. Up to the age of 5 years. This is done with vitamin A. Then after second was vitamin D. Vitamin D, D means, you can remember like D means Dharmendra sir. And bones of Dharmendra sir are strong. Now why I am saying? Because name of vitamin D is calciferol. And calciferol is associated with absorption of calcium. So absorption of calcium is triggered with the help of calciferol. So that's why whenever there will be lack of vitamin D, the body will face the problems of bones. For example, in kids, in children, there will be a problem named rickets. The bones will not able to bear up the weight of the kid. So the bones will be deformed like this. So the X-ray of anyone who is suffering with rickets will be like this. While in adults, it will not be rickets. It will be osteomalacia. Now what is osteomalacia? The bones will become soft. Bone is a connective tissue. And the beauty of bone is not the color of bone. The beauty of bone is the compact structure of bone. Within bone, there are two parts, hard part and soft part. The shaft of bone should be compact and hard. But in case of osteomalacia, the patient will suffer with soft bones. Always remember one more thing. If there is osteomalacia, it is associated with deficiency of vitamin D. But if it is osteoporosis, then it is associated with deficiency of calcium. From here, one more thing of biotechnology, the calcium deposition in bones is associated with physical fitness of anyone. That's why one test has been introduced by the Indian cricket team. And the name of that test is DEXA test. DEXA test. Yes, not only this doping test, but DEXA test is also. Yo-yo test is also there. Yo-yo test and DEXA test. Yo-yo test is to measure out the stamina of the patient. Not the patient, sorry, of the player. While the DEXA test is to find out the density of bones with the help of X-ray. Because bone density is directly associated with the health of the player. Then after, third thing you need to remember is vitamin D sources are not etc. etc. There are selective sources of vitamin D. First is sunlight. Whenever you will go in sunlight, sunlight will not give you vitamin D. But when your skin will came in contact with sunlight, the sunlight is carrying UV radiation, ultraviolet. There are 3 types of UV radiation: UVA, UVB, UVC. Not all the UV is absorbed and retained by ozone. The good UV is supposed to come down. That good UV, it will enter into your skin. And in your skin, there are cells named as melanocytes. These melanocytes will produce calciferol. So this is first source. Second source is egg. Third source is fish. Fourth source is liver meat. Why the meat of liver? Because any animal who is a mammal or who is a bird, if you are eating the liver of that animal, liver is storehouse of vitamin B, liver is storehouse of vitamin D, liver is storehouse of vitamin A also. Up to months, the body can run on the stored vitamins which are in liver. That's why whenever any person is alcoholic, he will face liver cirrhosis as well as definitely go for blood test. Any alcoholic will face, alcoholic means the one who is addicted to alcohol, will face deficiency of vitamin B12. Now next thing you have to remember is vitamin E. E is also named as beauty vitamin. E is also named as vitamin which helps in reproduction. The sources of vitamin E are all the dairy resources. Vitamin D is also found in dairy resources. You remember this thing. That's why milk is a complete food excluding two things. There are only two things milk is not carrying. One is iron, other one is vitamin C. Other than this, all the necessary things are in milk. But the milk, if you are drinking the milk of your Rajasthan's dairy named Saras, you can find out one thing on that food. There is a symbol named F, like this, and plus. If it is written on any food, that means it is fortified. Fortified means necessary nutrients are added by men in that food. So if the milk is coming from the dairy house, that milk is containing iron also. But if the milk is not coming from the dairy, or the local vendor is coming to your doorstep, always remember that milk is deficient in two things. One is vitamin C and one is folic acid. Iron. In the same way, there are fortified attas also. Now see, you have to remember one more thing. Vitamin K is directly associated with symbiosis. Symbiosis is a mechanism in which one organism in a plus-plus relationship with other organism. There is a bacteria named E. coli. Coliform bacteria are also in your intestine. So in large intestine, if you will go, then there are three segments of large intestine. One is cecum, one is colon, and third is rectum. Now in the area of cecum, there are bacteria, coliform bacteria and E. coli. They help you in digestion, as well as E. coli is source of vitamin K also. And vitamin K is named as phyloquinone, by the way. We are done with all the vitamins which are fat soluble. Ram Ram ji, Ram Ram, okay. Chennai se live, okay, good. Now, vitamin A, D, E, K. Other than this, there is vitamin C. C is named as ascorbic acid. Always remember C, vitamin A, vitamin C, vitamin E. These three vitamins are example of antioxidants also. Antioxidants are the nutrients which are helpful to reduce the oxidative stress of your body. Vitamin A, vitamin E, vitamin हिंदी मीडियम में कब लोगे? 2-3 दिन बाद होगा, देखते हैं. So vitamin A, vitamin E, vitamin C, these are antioxidants. Other than this, many of the minerals, they are also antioxidants. For example, zinc. For example, selenium. They are also antioxidants. Whenever you are talking about vitamin B, B1, B2, B3, B1 is thiamine, B2 is riboflavin, B3 is niacin. Out of them, only two are important by the point of disease. Beriberi is a disease which is associated with B1, while pellagra or 4D disease is associated with B3. B5 is named as pantothenic acid, B6 is named as pyridoxine, B7 is named as biotin. The deficiency of biotin can lead to hair loss. You can recall it like that. At 7 o'clock, my class was there and I am a bio teacher and I am facing hair loss. Then after B9, B9 is folic acid. We are talking this whole Ramayana thing about the B9. And then after there is B12, B9 is folic acid associated with the absorption of iron. B12 is associated with pernicious anemia. The name of B12 is, yes, absolutely correct. Hopefully your brain is answering. Name of B12 is cyanocobalamin. Cobalamin, it is carrying cobalt. So with this thing, within 5 minutes, we are done with all the vitamins. Now come back to this place. Now, whenever you are talking about sickle cell anemia, now in case of sickle cell anemia, the shape of RBC is deformed due to deformed hemoglobin. Now remember one more thing here. What is that thing? The deformed hemoglobin is actually a result of a genetic deformity. Ankita Jakhad, is it R छोटू? एक बच्चा था उसको मैं छोटू छोटू बोलता था, ये वही है? Now see, the deformity of hemoglobin, how to understand it? Now see, whenever you are talking about structure of hemoglobin, you have to say hemoglobin is made up of a central iron. Now always remember, iron is always present in +2 oxidative state. Now when it will bond with oxygen, because the function, okay, there are 4 chains also. This is the structure of hemoglobin. This is the molecular structure of hemoglobin. The central part is made up of iron, +2 state, that means ferrous. But when it will bind with the oxygen, that is the duty of hemoglobin to bound with the oxygen. So as well as it will bind with the oxygen, it will convert into Fe+3 state. So here is Fe+3 state. So now this is normal hemoglobin. Now these chains are named as 2 chains are alpha and 2 beta chains. Always remember 2 alpha chains and 2 beta chains. These are chains of proteins. Now these chains or the formation of these chains is governed by special type of set of genes. That's why in our body, the hemoglobin, our hemoglobin is named as hemoglobin A. Always remember, any adult is carrying hemoglobin, and that hemoglobin is type A. Other than this, there is hemoglobin type F also. Hemoglobin type F also. Now always remember these alpha and beta chains. These gamma chains. Not alpha and beta, alpha and gamma chains. Now these chains are responsible for formation of hemoglobin molecule. Now sometimes because of genetic deformalities, hemoglobin structure is deformed. Because of deformed hemoglobin, this hemoglobin will be named as HbS, and this leads to problem which is named as sickle cell anemia. Now what will happen with sickle cell anemia? On the junctions or on the dividers in blood vessels. For example, this is one blood vessel, and in blood vessel normal RBC will flow easily, but if the patient is suffering with sickle cell anemia, the RBCs will get stuck here and the patient will not be able to receive proper blood supply. One more thing you need to remember about RBC: human RBC is devoid of any type of nucleus. They don't have nucleus. One more thing you remember: even the camel's RBCs are also not having nucleus. Many times in many of the institutions, many of the teachers, by mistake, they speak or they teach that camel RBC is provided with nucleus. I heard myself that some student was saying that, sir, we heard that, and in today's lecture our teacher tell us that camel is having nucleated RBC. No. If RBC is not having nucleus, that means it cannot reproduce or it cannot divide itself. That's why RBCs are formed from the bone marrow. Within the bones, there is a hollow tube. Within this tube, there is a stuff named as bone marrow. Bone marrow is having red bone marrow cells. With the help of longer bones, the RBCs are formed. This process is named as erythropoiesis. Always remember, what is erythropoiesis? If someone is going to ask you, you have to say erythropoiesis means the process of formation of, yes, RBCs or new blood cells. What will happen if someone's body is not able to produce new hemoglobin? In that case, the patient will have a disease named as thalassemia. Always remember, thalassemia is also an example of genetic disorder. So someone was asking in the comments, thalassemia and sickle cell anemia, are they same? No, they are different. In case of sickle cell anemia, RBC is produced, but it is deformed due to the deformed chains of hemoglobin. This is also a genetic disorder. But in case of thalassemia, hemoglobin is not produced. Why hemoglobin is not produced in case of thalassemia? Because of genetic disorder, if hemoglobin is not produced, new RBCs are not produced. That's why the patient of hemoglobin thalassemia will have problem in his teeth also. Why? Because if hemoglobin is not produced, the body is overloaded with iron. So when the doctor will examine the hands of the patient will show deposition of iron and lack of RBC. So the patient's teeth will have a different color. The body will face two problems. One is lack of RBC, that means severe anemia, and second is overload of iron, because iron is not utilized anywhere. The entire amount of iron your body is having is X gram, then 70% of this, more than 70% of this is lying within the system named as hemoglobin, because the central part of hemoglobin is made up of iron. Ferrous, ferric, ferrous, ferric, ferrous, ferric. Now one more problem is there associated with hemoglobin, that is named as methemoglobinemia. Now what is methemoglobinemia? Because of nitrate poisoning or because of some other issues, if the patient, listen, because of nitrate poisoning, in Jaipur, nitrate poisoning is very common. Why nitrate poisoning is very common in Jaipur and in the surroundings of Jaipur? Because in Jaipur, printing industry is the major industry, especially in Sangane region. Now, all the industrialists, what they are doing, they are putting all their industrial waste into the water holes, which are actually prepared for recharging the groundwater. Now, these industries are using a dye named as azo dye, AZO, azo dye. Azo dye means cyclic structure with a nitrogen. Because of that, nitrate poisoning is a very common case in Jaipur and surroundings. Due to nitrate poisoning and many other issues, the hemoglobin of the patient will be available in ferric state. Listen to me, in a normal person, most of the hemoglobin will be in ferrous state. But in case of methemoglobinemia, the patient's hemoglobin will convert into ferric. Because of availability of this ferric state, the hemoglobin will not be able to bond with the oxygen. That's why the patient will face a lot of issues which will represent the symptoms that patient's vital organs are not able to receive proper oxygen. That's why this will represent characters like he is suffering with severe anemia. Now, whenever they are talking about any of the disease associated with hemoglobin or lack of oxygen, always remember one more thing: carbon monoxide. Whenever you will burn any carbon in the absence of oxygen or in the lack of oxygen, the carbon, whenever it is burned with proper amount of oxygen, it will produce CO2, carbon dioxide. But when there is lack of oxygen, for example, this is a closed room, studio is a very small room, and I started to burn a lot of papers or coal here, it will start to utilize the oxygen which is available in this room. As a result of this, this room will be devoid of any type of oxygen. When there is no oxygen, that means in lack of oxygen, the coal will burn and it will produce CO. CO is carbon monoxide. Carbon monoxide is carrying 300 times more affinity than the affinity of oxygen to bound with hemoglobin. Affinity means capability or love to bound with someone. So if hemoglobin is standing here, I am the hemoglobin, and on this side there is oxygen and on this side there is carbon monoxide. Firstly, I am supposed to go and make a bond with carbon monoxide, not with oxygen. Why? Because carbon monoxide is approximately 300 times more, you can say, capable to attract more the hemoglobin than the oxygen. That's why the patient will die. Then after, one more thing you have to remember about hemoglobin. Hemoglobin is made up of two parts. One is globin. Globin is protein. One is heme. Heme means iron part. That's why anyone who is suffering from the deficiency of hemoglobin will always be cured or treated, or the supplements which will be provided to him are actually the supplements of iron folic acid. They are named as iron folic acid supplements. In India, they are available free of cost. In fact, in India, Niti Aayog is preparing and publishing a ranking also, Anemia Mukt Bharat ranking, AMB, Anemia Mukt Bharat ranking. Now in AMB, one strategy is folic acid and iron supplement. Second strategy in AMB, 6 into 6 into 6. You remember 6 interventions. So one of the intervention is deworming. Deworming means albendazole, mebendazole. These are the tablets, these are the medicines which are given to patient when we want to kill all type of helminthic or worms in the intestine of someone. So that is deworming. 2 times deworming is performed by the government of India. That is named as National Deworming Mission. Deworming days comes 2 times in a month, in a year. So you need to find out nearest deworming day near to your exam. Then after, third thing you need to remember is they want to change the mindset of people. How they will change the mindset of people? Now understand, very important thing is delayed cord clamping. Now what is delayed cord clamping? Whenever the baby will be born, the baby is attached with the mother with the help of a cord named as placenta. So now doctors are saying don't cut the placenta immediately. Take some time. This is named as delayed cord clamping. This is also part of the 6 into 6 into 6 strategy of Anemia Mukt Bharat. And many other things are there. But whenever you are studying RBC, I think this much is enough. Then after, there is next thing, platelets. Okay, one more thing is here. RBCs. RBCs are having glycoproteins. Glycoproteins means proteins which are provided with carbs. Proteins and carbohydrates, they together are named as glycoproteins. Just for a second, you have to remember this thing. Now, these glycoproteins are responsible for blood groups. Whenever you are talking about blood groups, don't forget that till the day more than 40 type of blood group systems are available. But on your level, in case of general knowledge, you have to understand only one system, that's ABO system. ABO system was provided by Karl Landsteiner, Dr. Karl Landsteiner, a physician, and after his discovery, he was also awarded Nobel Prize in Physiology. In 1901, he discovered this ABO system, and in 1930s, I don't remember the exact year, in 1930s, he was awarded Nobel Prize for his discovery. Why it took 30 years? Because in 30 years, people came to know that this is the most useful blood group system out of 40 blood group systems. Now you remember one thing, ABO system is entirely dependent of 2 type of glycoproteins. One is A and one is B, named as antigen. So if anyone's RBC is carrying A antigen, it will be termed as A blood group. If someone's RBC is carrying B antigen, it will be termed as B blood group. If someone's RBC is carrying 2 type of antigen, it will be termed as, yes, AB blood group. And if anyone's RBC is void of any type of, you can say, antigen, it will be named as what? 0 blood group or O blood group. So A, B, and O. O is not having any of the antigen. AB is having both the antigens. B is having B antigen, A is having A antigen. This is very easy way to understand. So if I want to test your blood group, how I will test? Very easy. I will take some proteins which will react with A and which will react with B. Only 2 type of chemicals I will take. These are actually proteins. Now what I will do, I will take out your blood. This is blood sample. In this blood sample, I will put 1 drop of the chemical which reacts with A, and I will put 1 drop of the chemical which will react with B. So if your blood is reacting against A, that means your blood is carrying antigen A. And if your blood is also reacting against B, that means your blood is also carrying B. So in case of any person's sample which is reacting against both the chemicals, I will say your blood group is AB. But if anyone's blood is not reacting against both the chemicals, I will say your blood group is O. The process of this type of, you can say, testing is based upon a phenomena named as agglutination. Agglutination means protein will react with other chemical and it will form a kind of glutination. That's why it is named as agglutination. So blood testing is performed on the basis of antigens, and antigens are glycoproteins which are found on the surface of RBC. Now what about antibodies? Antibodies are opposite to antigen. So blood group A will carry the antibody anti-B. This will carry antibody A, but AB will not carry any antibody. Why? Because if it will carry antibody A or antibody B, it will attack on itself. O will carry both the antibodies. That's why O will not receive blood from A and B and AB. O will only receive from O. But O is not carrying any of the antigen. That's why O can donate to all of them. That's why O is considered universal donor. But after X years, Dr. Karl Landsteiner, with the help of Wiener and with the help of one more person, Alexander. 3 of them were there, but no need to worry about 3 names. You have to remember only 2 names, because even in the books up to MSc, they have not mentioned the name of Alexander. So you have to remember only 2 names. One is Dr. Karl Landsteiner, other one is Wiener. They have discovered in the jungles of Africa, when they were studying about the rhesus monkey, they studied the RBC. And there they have discovered one more thing. What was that thing? That there was one more antigen. And this antigen is named as, yes, RH. RH stands for rhesus factor. This RH is again, you can say, chain of amino acids. And on the basis of RH, again, these 4 blood groups are divided or bifurcated into the positive blood group and negative blood group. So if my blood group is B positive, that means my blood, my RBC is carrying RH factor, as well as my RBC is carrying B antigen. If someone is saying my blood group is O negative, that means on his or her RBC there is no antigen as per ABO system, and there is no RH. If someone is saying my blood group is ORH positive, that means what? He is trying to say his RBC is devoid of any type of ABO system or ABO type of antibody antigen, as well as, but his body is having RH protein. Now one of the most important question, which is favorite question of RAS exam, is RH incompatibility. RH incompatibility. Always remember, this will only happen when husband is RH positive and whenever wife is RH negative. Other than this, RH compatibility will not affect. Now remember one thing. As per Mendelian genetics, you remember Gregor John Mendel, working as father, priest, the famous priest who was having more trust in science, and he studied pea plant, and then after he studied Pisum sativum's 7 characteristics, and then he propounded laws which are also named as laws of Mendelian genetics. Under that, there is first law, that is the law of dominance. Dominance means if two characters, if one character is carrying, you can say, opposite type of, you can say, one is taller and another one is dwarf, when you will cross them, one of them will dominate. In the same way, whenever any couple which is RH positive and RH negative, any one of them is RH positive, one is an RH positive, then there are chances, I'm not saying 100%, but I'm saying chances that most of the children will be RH positive. Now, on the basis of this thing, we will understand this RH incompatibility. Now, see, here is the lady. This lady is pregnant. And in the womb, there is baby. And you know, this lady is RH negative. So the blood of lady is RH negative, but the husband of lady is RH positive. So I can say that baby which is in the womb of baby lady is RH positive. Now lady is RH negative. Now during the time of birth of baby, this RH positive will came in contact with the RH negative of mother. Now this is first pregnancy. No damage, no harm will caused, but one thing will happen due to this reaction, because the RBC of baby is having extra RH which is not available on the mother's blood. So the mother's body will respond against this, and the baby's birth will take place happily, but now the mother's blood will prepare antibodies. Antibodies against what? Antibodies against RH. So anti-RH or D antibodies, they are also named as D antibodies, or they are also named as Rahogam. Now see, this baby is second pregnancy. Now this time mother's body is having RH antibodies or D antibodies. Now again, if again the baby is RH positive, and because mother is RH negative plus antibodies, so this time this baby will face many problems. Many problems, like this baby can face anemia, this baby can face jaundice, and in some of the cases, rare cases, this baby can die. Now remember, in BSc and MSc books, it is written that baby will die. So if in exam they are going to ask you, you have to say, okay, the baby will die. मेरे को क्या लेना है, मेरे को तो क्वेश्चन सही कराना है यार, मेरे को कौन सा बहस करना है किसी से. आई एम नॉट हियर टू मेक एवरीवन करेक्ट, आई एम हियर टू एंश्योर दैट माय आंसर इज करेक्ट. सो इफ दे आर आस्किंग बच्चा मर जाता है, हाँ मर जाता है भाई साहब, मर जाता है. तुम कह रहे हो मर जाता है, मर गए सबके बच्चे, मैं भी मार के आ गया, कहते बात खत्म, एग्जाम सही, आंसर सही हो जाएगा तुम्हारा. In case of, if the baby is dying because of this process, it is named as erythroblastosis fetalis. Erythroblastosis fetalis. Fetalis is from fetus, and erythroblastosis means it is damaging the RBCs. Now to avoid this complication, the only thing is injection. Injection of what? Injection of anti-D. Now what is anti-D? It will stop the formation of antibodies. That's why it is named as anti-D. This injection is also named as Rahogam injection. RHOGAM, Rahogam injection. Again, I am going to tell you how they will form this Rahogam injection. With the help of the same technology which we have studied in today's lecture. Which technology? Recombinant DNA technology. Now, please type Y and allow me to drink some water. If you will type Y, I will proceed to the next segment. Now, next thing, what they can ask you from the chapter of blood, next is WBC. Whenever you are talking about WBC, you need to understand one basic thing. WBCs are part of your immune system. So whenever you are talking about WBC, there are two types of WBCs. One is when you will look them in the microscope, they will zoom out like this. One will zoom out like this. Other one will look like this. So these are granules. So from granules, you have to remember granulocytes. So one type of WBC is granulocytes. Other one is not having any granule, so it is named as agranulocyte. So there are two types of WBCs, granulocytes and agranulocytes. Now granulocytes, on the basis of dye from which they will take colors, they are divided in three types. One is basophils, one is eosinophils, other one is neutrophils. And agranulocytes, they are divided in two types. One is lymphocytes, one is monocytes. So there are five types of WBCs. One is basophils, two is eosinophils, third one is neutrophils, fourth one is lymphocytes, fifth one is monocytes. Now just imagine they are going to ask you which one of them is the largest WBC. So your answer should be the largest WBC is monocytes. Now there is a process named as phagocytosis. What is phagocytosis? Phagocytosis means one cell is eating up the other cell. That process is named as phagocytosis. Monocytes and neutrophils, they are involved in phagocytosis. Always remember this thing. Phagocytosis means I'm breathing. Whenever I'm breathing in, a lot of bacteria, a lot of germs are going into my body. Now as soon as these cells, neutrophils and monocytes, they will see any of the bacteria, any of the harmful cell entering into my body, they will start to act. How they will act? They will start to try to kill that thing. How they will kill that thing? They will eat up it. They will digest it. They will eat up it and they will end up this thing that yes, how you can affect me. I will not allow you to affect me. That's how they are defending your body. That's how they are protecting your body. Yes, they are kind of garbage trucks of the immune system. Now see, whenever you are talking about WBCs, you have to understand two or three more things. And then after you can say that you are done with what? WBC. Basophils. Basophils are having granules because they are granulocytes. Now within the granules of basophils, there are two types of chemicals. Both the chemicals are started from the H. Now one H is heparin. Other one is histamine. Heparin is formed in liver. Always remember, heparin is manufactured by liver, but after liver it will store into the granules of basophils. Now, what is the function of heparin? Because of heparin, blood never clots in blood vessels. Blood never clots in blood vessels because of heparin. If I am walking from here to there, the reason behind that is heparin. Heparin is anticoagulant. Anticoagulant means the component of blood which will stop the blood to clot. That is heparin. Then after, second thing is histamine. Histamine associated with a lot of things. For example, if someone had a, you can say, honeybee bite, then you might have heard that that person is so much sensitive towards a bite of honeybee or mosquito that he or she was in need to be hospitalized after this incident of bite. Why? Because of histamine. Whenever you are sneezing, it is because of histamine. So whenever you are taking any medicine which you are taking for so-called jukham or viral infection, or so-called sir dard, or so-called sardi jukham, and if that medicine is named as cetirizine, or a component of that medicine is cetirizine, that means you are taking a medicine to stop the histaminic action. So histamine is associated with increased secretions in your body, increased secretions from your eyes, increased secretion from your nose, increased secretion from your throat, increased secretions from the mucous membrane. Now see, any medicine which is named as antihistaminic, example is cetirizine, that will stop the action of histamine. This question was asked, I think, in last year's RAS, that histamine is secreted from which of the, you can say, blood cells. So you have to say basophils. Or there was some question about histamine. I don't remember the question exactly. Then let's talk about eosinophils. Always remember, eosinophils are associated with all type of allergy, especially if anyone is having asthma. That is associated with the action of eosinophils. Eosinophils, with the help of mast cells. Mast cells and eosinophils, both of them are responsible for any type of inflammatory response in your body. Anywhere if there is inflammation, that is because of eosinophil and mast cells. Anywhere if there is allergy, specifically the allergy of respiratory tract, for example asthma, that is associated with eosinophils. So we are done with all three type of granulocytes. One is basophils, heparin and histamine is important thing to remember. Eosinophil, allergic reaction and asthma is associated with this thing. Mast cells and eosinophil are associated with inflammatory response. And third was neutrophils. Neutrophil means they will eat up. They will eat up all type of bacteria, all type of germs which are responsible for, you can say, damaging your body. So that process is named as phagocytosis. Then after, let's move towards lymphocytes. Fourth type is lymphocytes. There are two type of lymphocytes, B lymphocytes and T lymphocytes. T lymphocytes stands for thymus gland. Always remember, thymus gland will work up to the age of 13 only. After that, it will start to shrink. If any one of you is below 15, and I'm in my, I'm around 40. So if, okay, so I'm around 40 and you, any one of you is below 15. Then if anyone will compare the thymus gland of you and me, then he will find out that my thymus gland is smaller than your thymus gland. So this is the only organ with age it will decrease in size, that is thymus gland. Now all type of T lymphocytes, they are produced by the same cells, same stem cells, but they will go in thymus gland and then after they will get matured in the form of antibodies. One more thing you have to remember, some type of T cells are very significantly important. For example, T4 lymphocytes, the decrease in the number of T4 lymphocytes is the primary symptom of HIV infection. I'm telling you in terms of GK. Now one more thing you have to remember, there are some T helper cells also. They helps the immune system. Lymphocytes are considered as natural killer cells, NK cells. NK cells or natural killer cells, they will go through your body, they will find out any of the cancer cell and they will kill it immediately. Then there are B cells. These are T cells. Then there are B cells. B stands for bone marrow. Remember, T lymphocytes are also produced by the same bone marrow stem cells. But B cells, bone marrow cells, are produced by bone marrow and they get mature in bone marrow also. Their function is also to protect, to defend your body. And the fifth and last one is monocytes. I told you, monocytes are associated with, you can say, phagocytosis. Then there are platelets. Platelets are also named as thrombocytes. The primary function of platelets is to help in clotting of blood. Remember that blood clotting is initiated by platelets. Fibrinogen will get converted into fibrin. And fibrin is a fiber-like structure. It will stop the blood. Then there is one more, prothrombin. Prothrombin gets converted into thrombin. Prothrombin gets converted into thrombin, and thrombin will convert the fibrinogen into fibrin. That's how the mechanism will work. In this entire process, calcium ions will play a critical role. That's why calcium ion or Ca+2 ions are also an example of clotting factors. And vitamin K is necessary for this process. If someone is carrying a genetic disorder named as royal disease, the meaning of royal disease is that person's blood will not clot. It can be because of deficiency of Christmas factor or because of deficiency of factor 8. There are two types of problems. Why it is named as royal disease? Because first of all, it was discovered in the royal family of Britain. This is genetic disorder, and the carrier of this disease is always X chromosome. Because X chromosome, we receive X chromosome from our mother. That's why it is said that mother is the carrier of this disease. This disease is named as hemophilia also. And always remember this, there is no treatment of hemophilia. That's why these patients are supposed to take injections of the factors whenever it is required. So I think we are done with blood groups. We are done with a lot of things from the biotechnology. And in case of any question you people are having in your head, you can ask me. Now see, during COVID, plasma therapy was quite, you can say, popular therapy. What is plasma therapy? The meaning of plasma therapy is you are supposed to take out the blood of the donor. From the blood, you are supposed to take out the plasma, because plasma is carrying all type of antibodies. You know very well that antibody is actually immunoglobulin. So antibodies are actually made up of proteins, and these proteins are made up of 4 units. These 4 units are actually 4 chains of amino acids. 2 are long chains and 2 are short chains. Always remember this thing. These chains are attached with each other with the help of disulfide bonds. So this is the structure of antibody. So these antibodies are present in a person who is, you can say, cured from COVID. So what they will do, they will inject this plasma into the body of patient. So the patient will receive antibodies directly. These antibodies were made by someone else. So this is an example of passive immunity. This is an example of passive immunity. So hopefully, I think for today it is enough. Any question if you are having, then you can ask me today or tomorrow. We will meet again tomorrow, and I will be waiting for your feedback. People who are in personal contacts with me, for example, many of them are our students. They can share their comments with me here or on the channel, or and good luck. Keep on studying. Tomorrow again I will pick up 2 or 3 topics, and we will try to cover as much as possible. Good luck to all of you. Keep on studying. Thank you.